I would like to invite Dr. Monica Sethi, Assistant Professor, Department of Biotechnology, Jamia Hamdard, to moderate the session. Thank you, Saima. To commence the session, I would like to invite our distinguished chairpersons, Professor Javed Ali sir and Dr. Shashi Kumar sir, to please come upon the dais and join us. Professor Javed Ali is Head Department of Pharmaceutics, Jamia Hamdard, New Delhi. He specializes in novel drug systems, including nanoparticulate system for brain targeting and oral bioavailability enhancement. Some of his prestigious awards include the Indian Pharmaceutical Association Medal, the SERC Fast Track Research Project Award, EST, the Career Award for Young Teacher, the Boycast Fellowship, and the INSA Teacher Award for Outstanding Teaching and Research. He has also been honored with the AAIPS IPA Distinguished Educator and Research Award in USA. He holds 5 Indian patents and serves on editorial board of prestigious journals like Expert Opinion on Drug Delivery and Current Neuropharmacology. Now, with immense honor, I would like to introduce our co-chair, Dr. Shashi Kumar sir. He is the group leader of Metabolic Engineering Laboratory at the International Center for Genetic Engineering and Biotechnology, New Delhi. His research spans metabolic engineering, synthetic biology, genome editing, and sustainable LD biofuel technology. He has held prestigious award positions at the University of Virginia, University of California, Berkeley, and USDA Albany. A recipient of multiple scientific awards and research grants, Dr. Kumar has delivered distinguished lectures on national and international platforms. With numerous high-impact publications and multiple patents, he continues to make significant contributions to the field of biotechnology and bioenergy. We welcome you, sir, and with this, I would now hand over the proceedings to our distinguished chairperson.
Very warm good morning to all of you. I think the session has already started, and the first speaker is the chairperson himself, Dr. Shashi Kumar, and I think Dr. Monica Safi has already introduced him, so I'll be requesting to deliver his talk. Thank you, sir.
Good morning to all. I'm Shashi Kumar. I'm from ICGB New Delhi, and I would like to thank from core of my heart to Professor Abdin for inviting this talk. I have been connected to the Jamia Hamdard from long time. I have worked in collaboration with Professor Abdin, Professor Faruqi. So it's a pleasure to come here again and again. Today the title of my talk is Mitigation of Carbon Dioxide and Climate Change. How they can be managed through the synthetic microalgae. If you see that, you have some. So if you see that every year 37 billion ton of CO2 has been released by by human activities. So 37 billion ton of CO2 has been released, and 50% is reversed through forest and algae in ocean and other fresh waters. So this way the 50% CO2 is remain in the atmosphere, making an extra layer in our atmosphere. So all the heat, sun rays coming, not reverting back to the atmosphere. So the earth temperature is getting up, increasing. So you can see the impact of temperature on climate. If there is a 1 degree temperature in Earth, atmosphere capacity increase to hold 7% more water vapor that cause heavy rains. You can see now in Himachal, Uttarakhand, it's happening and we're getting more rains nowadays. Uh, If there will be 2 degrees, then you can see that there's coral extinction will happen, glacier will be melting, river will dry after years, and this will trigger multiple climate catastrophes. Uh, as per science, there is already 1.5 degree temperature of Earth has been increased. And you can see the report, just one example in 2022, about 200 pilot whales die on the sea coast of Australia beach. And recently, yesterday, again 150 false killer whales stranded on beach in Australia for that. So, and if you want to push them back to the water, they don't want to go. They just want to die because of the climate, the environment getting so toxic. So, India has a net zero commitment, net zero carbon bioeconomy by 2070, as committed by our Prime Minister in Glasgow Summit of 26. And there's a like finance budget of total CO2 emissions. Uh, we have like 3 billion. We have used already 2 trillion tons. We will have to be the 0.9 billions now. And India is coming in the top five CO2 emitters. So it's important to. These are the sources of the carbon. Like in India, if we see 51% Indian electricity coming through the coal burning. This is called powerhouse or coal power burning plant. So having 44% global carbon emission, which is more than the vehicles that has been blamed, followed by like oil we use in our vehicles, machinery. Then government has introduced another that we should not import this gases for burning. We should produce through the this Satat programs, which will again produce the CO2 like 30 to 45%. So gas at 21%. There is steel manufacturing to produce 1 ton of steel, 1.8 ton of CO2 is removed. Even the buildings you live in, to produce 1 ton of cement, we produce 1 ton of CO2. So this all adding the climate change. So here we providing a simple solution. Algae, algae is a photosynthetic organism. You can say the simplest unit of plant which grow in the wastewater, need the waste CO2, and grow in sunlight can produce a biomass, and this biomass can be converted to nutrients or oil to fuel or biochar mixed with vermicompost can be used as biofertilizer. This like in Europe already like cement company they circulating all the CO2. There's zero CO2 emission from the cement industry to handle the cement. It's called algal carbon capture project in Sweden, and they observe that this biomass generated feed to the animals. Their properties in milk or egg or meat has been improved tremendously. Several other companies also working in the same direction. We also have been working with Chennai Petrochemical Limited Avan infrastructure in Chennai, and we have developed the synthetic strains which can capture the carbon double the amount than the wild type if CO2 is given to them. These are two marine algae were developed for them. Similarly, we have been working with Tata Steels. They also produce lots of CO2. So we have developed the synthetic strains microalgae for one, which if you see that wild type algae cannot sustain more than 3% CO2, whereas this synthetic microalgae can grow in a very well in the flue gas. Flue gas of industry contain like 10 to 15% CO2. They growing very well and producing biomass like 4 to 6 times more. And this synthetic strain thrive well in a high CO2. They don't thrive in normal air, so they are well protected. They are self-contained, do not thrive in survive, so they will not harm to the atmosphere. Similarly, there's another project we doing this to produce sustainable aviation fuel. Fermentation yeast produced 33% CO2, while algae take the CO2. Algae produce O2, so O2 oxygen is required by fermentation. So that way we can have like circular bioeconomy without producing any CO2 in the environment. So these algae are synthetic algae, which when you uh, uh, ignite to the blue light, uh, illumination with the blue light, these algae fatty acid change into the hydrocarbon, which is directly can be used as the biofuel for aviation fuel. So this is the overall that how algae we use them because you cannot produce one product to make it economically viable. So you have to produce several byproducts. So here these are the photobiological verticals where we grow algae. Then through this bucket centrifugation, we harvest this algae biomass separate from water. And through hydrothermal liquefaction, we can get the biochar and the biocrude oil. Biocrude oil can be refined through existing refinery to produce any kind of fuel. Biochar, we have seen that like in cellulosic ethanol industry, to produce 1 liter of ethanol, 15 liter spent waste is produced, wastewater is produced, which cannot be used for to grow anything. It is so because of melanite pigment there. But if you put this biochar in the column, the water gets clean, algae growing, plants growing, so it's a bioremediation of water you can do. And we have seen that biochar used with vermicompost can increase 50% yield in the tomato plants. So it's a good fertilizer also. Another work we working doing with space with ISRO and NASA, how we can provide the supplement food to the astronauts so they don't need to carry every time big load for the foods. And so because algae grow faster in due to microgravity, there is no gravity in space. They can grow in harsh conditions from last 2 billion years when there were no humans existence was on the earth. These algae were surviving. They grow in wastewater and with diluted urine, which contain the NPK and plant growth hormone could be useful for supporting the growth. They capture CO2 and give oxygen. They can provide sustainable food source having protein, carbohydrate, and many important antibiotic vitamins for astronauts, which will be staying for a longer time. So this is the simple process how we can use them. This is the one algae which produce astaxanthin. Astaxanthin is the pigment which make your muscles so strong. If you see the salmon fish eating these algae, they can swim against the against the water stream. So because of heating discussed earlier. So the problem which we solving in space is in the International Space Station agent. There's an issue that the level of CO2 in the ISS is 4.7% higher than the Earth has only 0.04%. So these astronauts consuming lots of CO2 and they always have had issues. CO2 level is being migrated in part impaired with various our organs and our psychomotors functioning, nerves functioning, our heart, our eyes get spread. So capture CO2 currently now they, through geolite they capture CO2 and through vacuum they release outside the ISS cabin in space. So they're making the pocket. So India also in 2035 they're on aim to settle their this this international space station, which will be called Bharat Antariksh Station. So they by 2035 they have already in four parts they're going to send it. One part they already sent in the space. So there will be three more parts will be sending them having more space by manufacturing. So it's for a new generation is the opening of new field. So here we provide the solution micro algae grow faster in microgravity than on Earth because on Earth algae settle down whereas in the space they keep on floating. And this is the perfect concentration which promotes the higher growth of algae. So this way algae can convert CO2 into food supplement, vitamins and release oxygen, reduce CO2 level in the cabin below 1%. And this is the procedure how we can use it. So with this, I would like to say thank you again to organizers for inviting me.
Thank you, Dr. Shashi Kumar, and very apt topic you have taken. I think we are going to take the question and answer at the last end of the session.
Sure.
Right? And the speaker of the discussion, next speaker of the session is Dr. Anjali Anand. Is here now? Yeah. Dr. Anjali Anand is a principal scientist and professor with the Division of Plant Physiology at the ICAR Indian Agricultural Research Institute, IARI, New Delhi. She earned her PhD from IARI and has specialization in seed physiology and climate change, contributing significantly to understanding plant responses to environmental stresses. She has been awarded with J.J. Chenoy Gold Medal for her significant contribution in plant physiology. She is a fellow of Indian Society of Plant Physiology. Professor Anand has guided numerous MSc and PhD students and has over more than 100 publications. She is going to talk on the topic physiological response of cereals to high temperature stress. Dr. Anjali Anand, thank you.
Very good morning, respected Chair, Co-Chair, the Organizing Secretary, Dr. Professor M.C. Dabdi, who is my senior from the student days, and he has inspired many of us with his diligence and dedication towards work. Throughout the student days, we have looked up to him and always wanted to be the sincere and dedicated student that he has always been. So he's a source of inspiration for many of us, and I thank the organizing secretary and the organizers for giving me a chance to speak before you here. So I have been working on high night temperature stress. So this night word is the key word here. Many of you would not have heard about night temperatures causing significant changes in the crop. So taking from where my predecessor, Dr. Shashi Kumar led, I'll also be talking about the impact of climate change with special reference to the night temperatures on the cereal crops. So these figures you must have seen appearing in the media, TV, and newspapers. These red colored areas all over the world seem to be very alarming as far as the temperature impacts are concerned. So when we segregate the temperature, when we talk about mean temperatures or average temperatures, we segregate them into the daytime temperatures and the nighttime temperatures. So what has been seen that the night temperatures have increased at a faster rate compared to the daytime temperature. And this asymmetry is growing. Now also you would have experienced in the last few days, and I think I read it in the newspaper this week itself, that the nights have become warmer than the daytime. So I have data from US uh, where they have shown that these about 50% of the areas have experienced more high night temperature compared to the areas which have been exposed to high day temperature. Coming back to India now. In India, we take up Kharif and Rabi season crops. So about 52.7% of the Kharif area and 54.9% of the Rabi area is under the strong influence of this increasing temperatures. And the past data it shows that the increment in the minimum temperature is about 0.28 degree per decade in the Rabi season and 1.19 in the Kharif season. So though these figures seem to be very less, if you see it as zero point 0.1 or zero point 0.2, but they have a high influence on the crops which are grown under these areas. So my topic will cover the high night temperature effect in the early sown wheat, and then I'll be talking about the pre-emphasis high temperature and post-emphasis high temperature and how it affects wheat as well as rice. And little bit I talk about the mitigation strategy, few experiments we have done, taking into consideration the application of ammonium nitrogen. So early sown wheat is also practiced in some areas of Punjab where rice wheat cropping system is there. And it is taken up because this cotton crop, which is grown prior to the wheat crop, that has to be terminated because of the infestation of bollworm. So now the farmer doesn't leave the field fallow for a long time, so he takes up early sowing of wheat to capture all the moisture that is present in the soil. But there's a trade-off between the night temperatures that are appearing around that time. So he has to sacrifice most of the germination. I mean, there's not a proper crop stand establishment during that time, so that has to be sacrificed by the farmer. So, but data is not available on what impact this high night temperature is going to have on the seedling. Plus, we do not have any donors to get it into the breeding program. So, one of my students, he phenotyped about 290 genotypes of uh, wheat panel. Under high night temperature, we have this facility where we can create up to 5 degree plus compared to the ambient. So after phenotyping, we found that around 41 genotypes, they had heat susceptibility index less than zero point 0.5. So these could be potential donors to be taken up in the breeding program. When the seedling characteristics were seen, then a significant difference was found in the dry weight vigor index, then canopy temperature, depression, leaf area, and first emergent leaf. All these traits showed a significant difference compared to the control condition. And by looking into the correlation matrix of the different phenotypic traits which he studied, a very high correlation was seen in the vigor index 2, that is dependent on the dry weight. So you can take it as you can interpret it as the varieties or the genotypes which have high vigor index, they will perform better or have a better crop establishment if grown under high night temperature conditions. Then the SNPs, GWAS analysis was done, and the SNPs were found to be located on chromosome 1 and chromosome 7. So next I will come to the vulnerability of the reproductive organs to high temperature. Here, most of the literature, if you see, most of the studies have been done with respect to the pollen because pollen is the part which is exposed directly to the temperature or dry air, while the ovary is shielded by many layers and it is within the flower. So the exposure to the ovary is less compared to the pollen. So, but reports are not there that what kind of vulnerability this is exposed to once it experiences the increase in the temperature condition. So another student of mine, she has done a very elegant study for which she was awarded a medal also. So what she has done, she has grown till the voting stage, and at 50% emergence, she has cut those ears and then she has taken those ears and cultured them under controlled condition. And the temperature she has given was +5 and +8 degree over the Indian condition. So then this was a detailed study, a very fine study in which she has removed all the pistils from the flower and all the pistils were sampled and further physiological and biochemical analysis was taken. The C14 was used to see where does this sucrose, C14 sucrose was used to see where the sucrose rose under +5 and +8 degrees centigrade. So these two varieties were taken. This is the sensitive variety, this is the tolerant variety. And in the tolerant variety, it was seen that most of the label, it was going in this part, which is the pistil part. But as soon as the temperature were rising, or we gave additional 3 degree, that is +8 degrees centigrade, then there was the distribution of radioactive sucrose changed between the pollen and the pistil part, that is the anther and the pistil part. Then she did the profiling of the sugars in the pistils, and interestingly, we found that raffinose has a role to play in imparting tolerance because the raffinose levels increased with the increase in temperature. So, like you would have heard of proline being a generic metabolite which accumulates under stress condition. As far as sugars are concerned, raffinose was a potential candidate for the stress response. The analysis of the raffinose synthase gene also showed that there was an increase in the transcript levels of raffinose synthase. Then some histochemical and quantitative measurements of H2O2 was done, which is the ROS. ROS is also again produced under all kinds of stresses. The ROS levels also increased under the susceptible variety as well as the tolerant variety. But if you see the scale y-axis, the y-axis here is only up to 200, but 3 times it is in the susceptible or the sensitive variety. So the homeostatic control is maintained by the peroxidase enzyme, which brings down the level of these H2O2 because the plant has to maintain the levels in the to further proceed towards the other metabolic processes. Analysis was done in the ADA and GA levels, and we found that there was an increase in ADA levels and a decrease in GA levels. So this gave a very significant lead to us because Paclobutrazol is a chemical which is used for lowering, for imparting tolerance or for getting the plants into the heat tolerant diet. And paclobutrazol, as you know, is an inhibitor of gibberellic acid also. So it gives a clear indication that these two are related. So this was a thermographic camera was used. So this measures the temperature of any organ or any part you are interested in to show how the organs are behaving. So temperature, thermographic temperature was taken. It is based on the infrared spectroscopy. So it was taken at 6 p.m. and night time and 6 a.m. So what it means is if you have less of differential temperature difference between the ambient and the organ temperature, that means that is a higher air temperature. If higher air temperature, that means all the processes which are going on in the developing brain can be affected. He found that in the sensitive varieties, the air temperatures are higher at the early morning hours. Early morning hour is the time when the flower is preparing for the pollination and the fertilization step to occur. So if temperatures are high, definitely they are going to have some impact on the floral biology. So finally, we proposed a multi-strategic response to in the tolerant genotype. So their strategy one is the homeostatic control of ROS, which we have discussed. Then AB and GA balance is disturbed in the ears. Then there's the rapinose accumulation, which is going to impart tolerance to the tolerant type, and of course the cooler ears, which which will lead to the metabolic process going on in the optimum temperature. This is another study in which the carbohydrate metabolism was studied in the spatially differentiated grains of the rice genotypes. Rice, if you have observed the flower, it will emerge and the lower ones will remain unfilled and the upper ones will be filling. So there's a spatial distribution between the filling of the upper and the lower part. So this study was done in two genotypes, sensitive and tolerant ones, under again increasing temperatures. Here we found that when this is the sensitive variety, here we found that the nocturnal respiration, that is a nighttime respiration, was rising in the sensitive varieties compared to the tolerant varieties. And we hypothesized that whatever loss in carbon is occurring because of consumption of this carbohydrate at night due to increase in respiration, that is made up in the tolerant variety by increase in photosynthesis. So there's some sort of a compensation mechanism that is going on in the genotype, which is helping them to be tolerant. So, according to our hypothesis, we found that there was an increase in the photosynthesis rate also in the Medina 22, which is a known tolerant abiotic stress tolerance, I would call it, because for drought also, heat also, it has a tolerant chemistry. What was interesting is that the inferior spikes in Vandana, they were not getting enough assimilate for filling. On the contrary, if you see, in case of Nagina, these red ones, if you see, they are getting the assimilase for filling. So this was also done through C14 studies where we could see the partitioning between different organs. And then this is the sucrose synthase activity which supported it and which showed that peduncle was acting as a bottleneck for the movement of sucrose from the primary to the developing brains. This is also recently published, just 15 days back, it was published in Plant Stress, this paper. So in this, we could profile all the sugars and we saw that this assimilate partitioning, what is happening, that inferior spikelets are filled in the tolerant variety while they are not filled in the sensitive variety. So this partitioning is being effected in the early morning hours of the night, or I would say late part of the night, or you can say early morning hours of the day, compared to the rest of the day. So the strategy what goes on in the tolerant and the susceptible genotype is that the tolerant one has a higher carbon pool because of the higher photosynthetic rate, higher sugar translocation, while the flowing which we showed through the sucrose synthase activity, more phloem unloading, and significant increase in the test weight of inferior genes. That was a remarkable finding which came out that all the grains are more or less equally filled in the tolerant variety compared to the sensitive variety.
Madam, please widen in the next.
Only two slides.
Okay.
So this is the relative change in the amino acid pool which we found because of we thought that foliar replication of nitrogen at the 5 days after enthesis may help to prolong the photosynthetic process, which otherwise was senescing because of the increase in temperature. Here we found that there was a variation in the number of amino acids and glutamic acid, glycine, and all these mentioned here, they were showing an increase under high night temperature. So this showed that the aspartate metabolism is the one which is affected, and all the amino acids which are originating from the metabolism of aspartate, they are acting as a respiratory substrate for the nocturnal respiration. So these are the findings. Again, I have listed out just for a recap of the studies I have shown. So finally, I would like to acknowledge my director, joint director, head of the division, and all the co-workers and my students who have contributed to this work. And in the end, again, I would like to thank the organizers for giving me an opportunity to present my work here. Thank you so much.
The next speaker of the session is Dr. Shivani Nagar. Dr. Shivani Nagar is a scientist at the Division of Plant Physiology, ICAR, Indian Agricultural Research Institute, New Delhi. Specializing in abiotic stress physiology, her research focuses on understanding plant responses to environmental stresses, contributing to the development of stress-resilient crop varieties. She has actively published in reputed scientific journals, advancing knowledge in plant physiology and stress adaptation. She serves as an editorial board member for Food and Scientific Reports, reflecting her commitment to the scientific community. She's going to talk on the topic understanding the role of plant APP2C gene family in abiotic stress tolerance and ADA signaling in rice. Dr. Shivani Nagar. Thank you.
Respected Chair, co-chair of the session, eminent scientist in the hall, and dear student and special respondents. A very good morning to all of you present here. I welcome you all to in my presentation on understanding the role of KRA protein phosphatase 2C gene family in abiotic stress tolerance and ADA signaling. So I myself, Dr. Shivani, I have recently started working on utilizing the CRISPR-Cas9 genome editing technology for understanding for improvement of understanding on every signaling pathway in rice. So I will. So we all know that whatever vegetation we see either in desert or in rainforests, they all evolved from the single cellular green organism. green organism, alga, to in the course of 500 billion years. So during this course, what happened was that there are few processes which had the key contribution were like sexual reproduction, instrumental regulation, UV protection, desiccation tolerance at cellular level and at plant level. Then vascular tissue development, development of complex cell wall and stress signaling and sensing. So these processes played a key role, and among these all these processes, if we see that ADA, abscisic acid, is a plant hormone which played a major role because we all know that colon viability during the seed desiccation, development of seed desiccation tolerance, and then stomatal regulation, all these processes are regulated by abscisic acid in plants. So this ADA, which is commonly known as stress hormone, it played a crucial role in enhancing plant adaptation. First, to adopt plants from marine habitat to land habitat, and later on, the sensing and signaling pathway kept on evolving to improve the abiotic and biotic stress tolerance in plants. We all know that ADA level regulates the stomatal closing and opening by regulating the potassium ion channel in by regulating the potassium and chlorine ion channel in stomata. So the influx of potassium and chlorine leads to the change in water potential and closing and opening of stomata. Then the ADA level also determines the activation of antioxidant defense system and the level of stress proteins like GIA and dehydrin, and also the desiccation tolerance by increasing the quantity of osmoprotectants in soil. So along with that, as the water uptake, roots are very important organ. So the root number, root volume, it is also regulated by the level of ADA in cell. If I talk about ADA biosynthesis and signaling, so ADA biosynthesis starts with the beta carotenoid and the formation of zeaxanthin, vinyloxanthin, which leads to the formation of ADA aldehyde and ADA. So under stress condition, what happens is that the two enzymes, which is zeaxanthin epoxidase and 9,6-epoxy carotenoid deoxygenase. So this ZEP and NCD, these two enzymes are upregulated, which leads to the increase in ADA level in cell. And this ADA is perceived by ADA signaling pathway. So ADA signaling pathway, the discovery of ADA signaling pathway was very important, and it consists of the ADA receptors, which is also known as pyruvate resistance 1, and pyruvate resistance like protein and RCAR protein. So these they are the receptor of ADA, which interacts with protein phosphatase 2C. So here you can see that in protein phosphatase 2C, in the absence of ADA, they are binding with the sucrose non-fermenting related ligand, and the pathway is blocked. So the protein phosphatase 2C is a negative regulator of ADA signaling pathway. And in the presence of ADA, so the protein phosphatase 2C is now able to bind with the receptor, and it releases the SNRK to activate its downstream to allow it work and activates the expression of ADA-mediated genes. So these ADA-mediated genes are related with the biosynthesis of various desiccation tolerance-related proteins, stress tolerance-related proteins, and other things which I talked about in the previous slide. So if we see the evolution of ADA signaling pathway, so it was not like the what we saw in the last slide. So the signaling pathway was also evolving as the plants were evolving from the marine habitat to the land habitat. So we can see that in alga, chlorophyll alga, there was the protein phosphatase 2C, SNRK were there, but ADA was not able to interact with the PP2C and SNRK. So as the habitats were coming, were evolving, so then the evolution happened where the receptors came in picture, but still the receptors were not able to interact with ADA. So it was in bryophytes for the first time that ADA receptors first interacted with ADA and then it interacted with protein phosphatase QC and all. So all this leads to the evolution of, and this signaling pathway also evolved in terms of increasing the number of various intermediates like receptors, protein phosphatase, and SNRP. So in case of rice, there are 30 receptors, more than 100 protein phosphatase gene, and 47 SNRP proteins. So you can just imagine the complexity of the signaling pathway of ADA. And we know that protein phosphorylation, which consists of protein kinases and phosphatases, which leads to the phosphorylation, and phosphorylation of any target protein is one of the most important pathway in regulating the activation and deactivation of protein function. So among these protein phosphorylates, the protein phosphatase 2Z, which is important regulator of like a family, which plays important role in ADA signaling. So the clade of protein phosphatase QC in Arabidopsis, it was first time was reported that protein phosphatase QC, the mutation for ABA1, ABA2, which is the PP2C gene. So these ABI1 and ABI2, if you the mutant was found, which leads to the insensitivity of mutant. So here we can see that in the presence of ABA, there was inhibition. The mutants were able to germinate, whereas the wild type was not able to germinate in the presence of ABA because we all know that ABA inhibits the germination. Later on, when these mutants were further studied for their stress tolerance ability, so it was also found that mutants have higher water use efficiency, higher water use efficiency, and low stomatal aperture. So overall, the water loss was uh, less in mutants. And similarly, it was reported in few rice reports also. So like this, for protein phosphatase QC, there are many reports which suggest that this gene family, these genes have important role in ADA signaling. So what we have at our lab that we are using the CRISPR-Cas9 genome editing technology. So the technology have a Cas9 protein which is like for which we have a guide RNA. Guide RNA helps to direct the Cas9 protein to a target site in DNA, and we can create a mutation at target site. So this technology, we are using this technology to generate the mutant lines for the clade A gene of protein phosphatase QC gene family, which plays role in stress tolerance. So, generation of these mutant lines will lead to a better understanding of all these genes in protein phosphatase AB signaling. So we hypothesize that if we inhibit, if we generate the mutants of this gene, what will happen that mutant will become hypersensitive to ABA and they will be having the enhanced stress tolerance. So for this, just a workflow of the technology, that for this we need to design the guide RNA. These guide RNAs are cloned in guide RNA expression cassette, and the binary vector is gamma. And then we generate the transgenic line which express the Cas9 protein and gRNA expression, gRNA cassette also. And the transgenic lines are screened for mutation, and then homozygous mutants are later on found in next generation. So now I will elaborately present the results of one of the mutant line, which is PP2C10. So this for this particular gene, we have the three mutant lines. So you can see here that as compared to the wild type, there is insertion of 4 nucleotides, and here there is deletion of 1 nucleotide over in this particular case. So the chromatograms are clear, which is like ensuring the stable mutation. These homozygous lines, when when they were their protein alignment was done, so this was like when the protein structural modeling was done, we found that this as compared to wild type, the non-functional protein was formed on mutation. So these wild type lines, when we got the wild type lines, when we evaluated for ADA sensitivity. So we found that in seed germination there was no effect in mutant lines. But at early seedling growth, we can see that there is a mutant lines have inhibitory effect. Then the seedling was tested for stress tolerance ability, osmotic stress, and salinity stress. So we can clearly see that two mutant lines in L2 and L3, in L2 and L3, that they were showing very good tolerance to the salinity stress and osmotic stress during the recovery phase. And when these seedlings were physiological studies were done, so we found that these seedlings, mutant seedlings, had better membrane stability, lower lipid peroxidation, lower level of have higher level of antioxidants and better seedling survival rates. So the level of antioxidants can also be seen that from this picture also. And at later stage during recovery phase, the growth rate of mutant seedling was very high. This was done under seedling stage when, and then further these lines were also evaluated under field. And the field also, although it was a single gene mutant, but we found a prominent growth inhibiting effect of single gene mutant in case of both lines under irrigated condition also and under drought stress condition also. And the mutant line shows the leaf rolling faster than wild type and less water loss. Further, like when these mutant lines, when they were evaluated for under drought condition in field, here we observed that growth inhibitory effect, and but the overall yield and yield was not affected. So which means that although there was decrease in biomass, but somehow the stress tolerance in plant was able to make up the loss in grain yield. And these are the thermal image pictures where the green, the first line is of wild type plants, and later on we have the mutant lines. The mutant lines were able to maintain their canopy cooler than wild type under drought, under field condition under field condition. So after this, we also have some few more mutant lines for other genes, which for them the work is still ongoing. So this is for 2212. We have the mutant lines, and then these lines, when they were evaluated under irrigated condition, we can see there is no. They are almost similar and under similar to the wild type. And in case of plant height and other growth parameter, also their behavior was same. Then we have the mutants for other genes like protein phosphatase UC8. So these lines also, but these lines show better growth. Although, as in case of UC10, there was inhibitory effect, but here we found that there was increase in biomass and also yield under irrigated condition in these mutant types, which was very prominent, that increase in biomass and in grain yield. So with this, we are in the process of generating some double mutant also by making the cross of single mutant. So overall, from the studies which we have done till now, although that protein phosphatase 210, which was a single gene mutant, but we can see that this gene have no role in ADA sensitivity at seed germination, but it plays a role in ADA inhibitory effect at early seedling growth, and also plays role in the stress tolerance at seedling stage, have growth inhibitory role in adult plants. And so these kind of generation of this kind of genetic material will help in improving our understanding about protein phosphatases, which are a very important part of ADA signaling pathway. With this, I sincerely thank Director IRI, Joint Director Research, and our mentor, Dr. Vishwanathan sir, all the our head of the division, professor, scientists, and my faculty members, and all research scholars who are working with us in our lab. Thank you all.
Thank you. Thank you very much. In this technical session now, we have one plenary session as well, where Dr. Ajit Kumar Shastri is going to talk on the topic plant immunity for sustainable production with reduced pesticide use. It gives me immense pleasure to introduce Dr. Ajit Kumar Shastri. Dr. Ajit Kumar Shastri is a distinguished scientist in plant biotechnology, molecular biology, and genomics, currently serving as Director of CSIR, NBRI, Lucknow. Previously, he led ICAR and IDB, New Delhi. Dr. Shafne's pioneering research includes India's first medicinal plant genome sequencing of Holy Basil, Osimum sanctum, along with significant contributions to menthol, mint, artemisia, and Osimum genomics. Let the audience know about you, sir. And his work extends to rural development and farmer welfare, leading initiatives like the CSI Arma Mission, and he has received numerous prestigious awards, national awards, and so on, with a commitment to cutting-edge research in society impact. Dr. Shastri continues to drive advancement in plant biotechnology and agricultural innovation in India. Dr. Ajit Kumar Shastri.
Ajit Kumar, Dr. Javed Ali, Dr. Artin, and my friend Anjali ji here from Plant Physiology. Thank you, Dr. Abdul, for inviting me here. And it's a wonderful atmosphere. So what I will advise you, so you can sleep because it is dark here. So if I say something, if you are not able to understand or I am not able to understand, you can sleep. Very nice atmosphere. So, but sleep with your eyes open because I can see you in the dark whether your eyes are dangling or not. So this is, I'll tell about the internal immunity. And what I will do today, I will give a concept. How much time I have?
30 minutes, like 25 plus 5.
Okay. I'll give you a concept. Concept, that's the internal immunity of the plant. Before that, I'll talk about my institute, NBRI. So the mandate is the mission undertaking basic and applied research. It's okay. 2 minutes, you will give me answer.
Yeah.
So the prospection of genetic improvement, particular emphasis on underexploited plants, non-traditional and wild plants. So when you go back to the non-traditional, now you are listening a lot about genome editing and many more things coming in the future and all the techniques. But we have to go back to the wild types. Why? I'll tell you. We have the focus area like floriculture mission, conservation of threatened plants, underutilized plants and bioprotection, climate resilient plants for indoor pollution abatement, arsenic risk assessment, natural product development, Indian local genetic improvement in plant resources. So we have the department starting from the botany, basic botany, taxonomy. So we have to identify the plants, bioprospection, utilization of the plant, but in between there is biodiversity, conservation, environment, bioremediation, again molecular biology. And it comprises of all branches of agriculture in addition to the basic branches. So that nowadays, seeing the climate change, every seminar you go, there will be some people at the other, they will be talking about the climate change. So this is a buzzword now, climate change. We are actually experiencing what climate change is, and we are listening to people what climate change is. So what actually we are doing. If we are doing research, we are doing it as single subject. But whether we are doing completely in an integrated way, that my institute does, taking care of the environment, soil, wire, and water. Do you know that? Well, you require 1.75 kilogram of oxygen every day, and one plant produces 1.2 kilogram every day. And what is the average of uh, plants actually required to remain alive in this world? So these are many things you have to think about. And when you go for the underutilized plant, they are the plants which are in the jungle. You are not caring about it, and you have to care now, right now. So having said so in the floriculture missions, we have over 29 states we have covered. What do we do? We go to the farmer, prepare a cluster there, and in the cluster, we give the farmer the planting material developed by NBRI, improved planting material, agricultural material, and handle them to go to the market. And also, we have pupillizable things like micro-circular economy. Suppose there is a temple or other garden. So there is flowers are required there. So create cluster all around that, micro clusters, 20 to 25 farmers. The flowers will come to the temple of the Daga, and then it will come out. Then when it comes out, then you have to prepare the fragrance out of the flower, the color out of the flower, the dhupati, agarbatti from the flower. So these are small things, but it means a lot to the common people. So when you do that micro circular economy in a small area, how many temples and dargahs are in the world, in India? Lots, huge amount. So can you improve the livelihood of those people in that area? That's our thinking. For that reason, we have a mission project called Agriculture Mission Planning. And if you go back, when we are actually starving, we are getting some food from USA, some rotten food, wheat, and we are waiting for that. At the time, the cropping area of India, now the cropping of area of India, it remains similar. But our productivity has increased several-fold in carbohydrates, rice and wheat, not in oil, edible oil or pulses, proteins of edible oil. So it has increased. How? We have utilized lots of pesticides, lots of with the plants, we made the plants to respond to the fertilizers. The genes are like that, they respond to the fertilizers. And we have increased our productivity. And by the way, the breeders of that period of time, they started breeding plants to increase yield because yield was absolutely necessary at that period of time. So everybody, and a breeder is a good breeder, when he see only one character, yield, and yield, and all other characters around it, the vitamins, the micro elements, the secondary metabolites, we forget. And we have forgotten. The taste earlier you were having, you might have listened from your mother and grandmother. The food, the taste it was having. Right now we don't have any food, any taste. Now what we have done, we have converted to fortification. Fortification. The term was called fortification. We will fortify it. We will fortify it with different micronutrients. We will put what is the micronutrients? AHPK, CAMBS, FAM, and ZSU, BOCL. And some of the micronutrients we require. We will fortify this with the micronutrients. And these micronutrients, we give it from outside. Now, agriculture, we are working on fortifying by breeding only. And there we are thinking about our wild types. And we are getting knowledge from our wild types. Right? What is there in the wild types? Now what is not there in the crop we have developed. So that, and in the process, when we have the plants which are deficient in micronutrients, vitamins, and secondary metabolites, we found those plants are susceptible to many insect pests, susceptible to abiotic stresses. The earlier plant, the wild plant, they are not susceptible, but now they are susceptible. Again, we have destroyed the soil, we have cultivated in a different way, not in the natural way. Thereby, and we are enjoying the environment every day. Our lifestyle is different. Suppose you are outside, it is raining, you will run away and stay inside the AC room. Plant cannot do that. Plant has an internal immunity that you have to recognize. That concept I want to give you. Plant has an internal immunity to stay there and face all the stresses, all the attacks. But we have a different immune system. We have studied from our childhood, IgM to IgG. There are 5 types, you know that. And there are many other surrounding factors. Phagocytes and CD4. Many people, IgG people teach you in many ways. So, but the plants, they don't have all these type of immune system. They have a immune system which is integrated in terms of vitamin, minerals, as well as the secondary metabolites, which gives you taste ultimately. So having said so, the climate change again they are facing you, the plants are, and our constitution what it says, we have to provide nutritious food to our citizens. This is directly principle. So we have the responsibility, scientists are here, students are here, we have the responsibility to feed our ever-growing population in future with nutritious food, not the food, the rice, white color rice, which having no taste, no smell, nothing. Again, I will tell another thing. Plants, every plant, they have aroma, you have a nose to smell it. Every plant, plant talk, you should have a ear to listen it. Every plant has immunity, you should have a immunity to look at it, discover it. So these are the things, these are the things we actually have. Before that, going to the immunity, we have synthetic. In breeding, the synthetic are well defined. Two genes together, coming together again and again, generation after generation, they are linked to one chromosome, the synthetics. But in case of the synthetic, molecular synthetic, what I am telling, this is the molecules which are present in one plant, also present in many other plants. I will give you example. The pathway, if the molecule is there, then pathway must be there. So the pathway is also synthetic. The molecules are synthetic, the pathway are synthetic. So this is example. Very small, normally, don't take it in mind. This one. So for 370 beta oxygen. If I take 1,000 plants, beta oxygen is found in 377 plants. That means this is synthetic. The pathway is synthetic. Again, the other compounds, lilandol, many other compounds. So there are many compounds in this. This is in the terpene pathway. And this is in the phenyl propagate pathway. Like this fumarins, stilbenes, catechins. So you can find out the molecules, little bit boring, sleepy. Half of the people are there going to sleep. Okay. Come back. So these are this is phenyl propagate pathway. I told you, don't take the pathway into your head. Just listen to me, you will get the concept. So, rosmarinic acid 57 plants out of 1,000 plants means the pathway is there, synthetic. And the line, this red line, of pairs, these are primary metabolites, these are secondary metabolites. And the name of the plants, monoterpenoids produced by these plants, sesquiterpenoids produced by these plants, like the triterpenoids, alkaloids, phenyl propanoids. Do you think a plant producing this particular chemical doesn't produce other chemical? Sesquiterpenoid? A plant producing monoterpenoid doesn't produce sesquiterpenoid? Not correct. So the plants producing monoterpenoid, you tell them the plants are dominant in producing those particular compounds. Sesquiterpenoids are there. The other compound diterpenoids are there. But how much amount? Why they are producing? How they are producing? That's the question of research. So we have to find out as a researcher further. And we have to correlate them in the environment, to the environment. The same species plant in Delhi, growing in Delhi, and growing in Chennai, and growing in Assam, will be producing different type of cover in different depot. So there is the effect of environment also. If the molecules are synthetic, the biochemical pathway is there, and they will produce different compounds in different places depending upon the environment. Sometimes before we are learning about the signaling pathway, how complex it is, only was things. So we have to find out why they are producing what these compounds are required for the plant at that particular region to fight for themselves to survive. They are in these stringent conditions to ward off the insect pest or to attract the insect pest. So like this, these compounds are absolutely required. So in CIMA, I was in CIMA earlier also. In CIMA, there was a last during COVID time, there was a locust attack, if you remember. So all plants they die except the aromatic plants. So there must be something in those plants which are actually repelling those insects, the locusts. So then, having said so, all the plants they die, acacia is surviving, the lemongrass is surviving, all other traditional plants they being eaten away. In the harmony of the plant, so these are the compounds. So plant in its harmony, contain this compound. These compounds are the plants, and we are very intelligent enough, and we utilize those compounds for our benefit. How many people were taking kadha at that period of time? How many people were taking kadha? Kadha from the plants, and these contain these compounds. So these compounds are we are utilizing for our immunity, and these plants they are producing those compounds for their own immunity. Some examples I will give, maximum examples from our own work. So there are some insecticidal activity and antibacterial already described in literature. Again, another, this is not our work, somebody else's work. If you increase the 6-hexanol content in the plant, then it will be producing cellulose, and cellulose will give tolerance to the tomato yellow leaf curl virus. So this is how it is giving tolerance to the. I will not tell the word resistance because absolute resistance is very difficult. Okay. Another literature. This is isoprene and alpha beta kinase through salicylic acid pathway provide resistance to bacterial infection. Similarly, beta carboxylase, jasmonic acid pathway. We all know what are the pathways in the pathway. So this pathway it gives tolerance to the plant. Okay. This is our work. This is in Udania. In Udania, if you play with different metabolites like subudamolites, minor udamolites, it will give tolerance to pseudomonas. So this is again another what I will tell. This is in potato. Again our work in potato late blind. So if you increase the fermentation content in the plant by inducing that gene, then definitely it is going to provide the late blight disease tolerance to the plant. Again, this work in NIPP when I was there, Srivastava, Rohini. You must be knowing Rohini. With her, I told to work on internal immunity. So what we did, Cardiac Kajal, it is susceptible to pot. So pot destroyed. But Cardiac platycarpus, which is the wild type, it is tolerant to pot. Then we analyze the genome of the platycarpus. Cardiac genome was known. Then you will compare. There are so many differences came, and from there, some of the clue we have taken. Many compounds we tested for the pot by shortlisting from the genome. What type of compound? Also, metabolism we have analyzed. And from there, we could find that epicatatin 3 galley, it's the culprit. It is more in platycarpus and less in cardiac standard. So thereby we took to the pathway. Okay. F35HG. This is in the pathway of the epicatatin 3 galley. This is the crucial gene. We found this gene is defective. Otherwise, the promoter is having higher number of MAB elements in the platycarpus genome. So thereby, this expression of this epicatatin 3 galley is maximum in Cajanus platycarpus in comparison to Cajanus carpus. For that reason, it is compared. It is not that it is driving away the insect; it is killing the insect. How? This is my elements. Yeah. Again, to transport to tobacco to different experiments, I am not going into those experiments. But if the epicatatin 3 galley जल्दी नहीं आएगी. Okay, then what happened in the insect? What happens if you feed epicatatin 3 galley or the tobacco leaf when the gene is being expressed? What happens to this? There is lipogenesis, decrease in lipogenesis and increase in lipolysis. we study the insect, what is happening. So this insect is being killed in Pajanus platycarpus, not in Pajanus caja. What is it? The secondary metabolites, they act for the internal immunity of the plant. Again, this is again our word is building plural. A small molecule in mentha, when you increase the concentration from 0.5 to 25%, the plant became tolerant to the leaf eaters. They are hairy caterpillars. Again, another example, Artemisia annua. So what we did, we stopped the expression from cinnamic acid to fumaric acid. When cinnamic acid being stopped, this is phenyl propionate pathway, then its higher amount of cinnamic acid goes to benzoic acid, salicylic acid, then produce artemisinin. Then when it does that, but the lignin biosynthetic pathway becomes defective. So the compounds, the fumaric acid will not be available for the lignin biosynthetic pathway, and this is very much visible in the xylem and flower. And the plant becomes susceptible to aphids. And aphids were earlier, the plant was not host to the aphids earlier. So now it became susceptible to aphids. Not only that, they became aphids and many fungi also. What we did, we expressed the cause, vinyl lineage. And we overexpress this so that fumaric acid earlier or cinnamic acid, after that cinnamic acid, fumaric acid not being produced.
You can change it.
This one. What will you do? Changing slides. Okay, I have to use steel. So earlier we were stopping here so that cinnamic acid was more. Now we are overexpressing part so that cinnamic acid more. But this downstream pathway will not be detected. And for that reason, so there will be production of again the plant will be alright, but artificial production will be higher. And now the insect pests and the fungi which were earlier attacking the plant because phenyl propionates were not there, now become tolerant to those insect pests and fungi. So having proved so, I think I'll speak this, you have less time. I'll just give you how we are using it. So this is the genomic resources we have right now, different aromatic plants. So and we have cloned some of the genes, and individually we are checking some of the genes, traffic genes, how they are actually helpful to your cool like clone genes. Individually we are checking. And these are the genes, how they are actually helping for the internal immunity of the plant. These are sesquiterpene genes. And see the amount of molecules in vetiver. Vetiver, you know, vetiver cuscus. Drink is there. During the summer season, you will drink that. So that group contains that many molecules. And believe me, the plant is tolerant to water stress, water bugging stress, salt stress, cold stress, heat stress, whatnot. Anywhere you grow the plant. So the molecules are responsible. We believe it. And we have to prove it. Another, the last sentence, we know we are having, I can categorize all of you three types, Vata, Pitta and Kapha in Ayurveda. In Ayurveda, there are three types of individual, Vata, Pitta and Kapha. So we work on that. And when we talk about the plants, Vata plant, Kapha plant, Pitta plant, the plants which actually cure those properties. And based on that, we can devise the plants medicines. So how actually this work? They are from different genera, different species, different family, but still they cure the same character. Vata character, Vata, Pitta and Kapha character. A person may be having 90% Vata, 60% Pitta and 30% Kapha. It is a very complicated thing. So how these plants have been used? So we work on that. So we know now 90, around 120 compounds are being from the 90 plus, they are being already available in the allopathic system. We selected the plants for Batasamanam, Pitasamanam, and Kapasamanam, and we did a lot of genomic analysis, but we could not find anything. But we published a paper, tissue specific expression, AFLP, we were going for the total sequencing, and some of the novel proteins, some of the proteins we have identified which can be studied in future, but we have done it. But when you analyze the metabolums, so then we analyze the metabolism of Bhattasamanam plants, we extracted, analyze the different compounds. जल्दी नहीं जा रहा है, मैं जितना जल्दी बोलने वाला हूँ उतना जल्दी नहीं जाता है, तो इसीलिए थोड़ा सा रुक जाता हूँ. So what I did, I asked 20 Ayurveda all over India, which plant you will rate highest and how in the scale of 1 to 10. So which one is first and which one is the last. Like this, we took the average. So 3 plants we could get: Google Pomifera for Bhattasamana, the Ricinus families, and Crucial Lancelota. And interestingly, if you analyze, they contain a triterpene. And triterpene, individual up to individual, I cannot go. But triterpene, in steroids are given to the people who are suffering from back arthritis. So there is a correlation. And specific compounds we will find out in the future. For this only I could find out, our group could find out. Then lots of analysis, then we are not going to that. How they are related to each other, which are the compounds. Then similarly, Pitasamanam. Pitasamanam, again, there are some glycosides. But still then we have to work a lot on that. How the common molecules can be identified and can be utilized in future, so that this can be authenticated. Then Papasamanam, phenyl propionates, gingiberine, and many other compounds like phenyl propionates, they are mainly responsible. So like this, so lots of analysis we have done. What I need to say that there is internal immunity of the plant. I try to give a concept to you. In future, we are going towards traveling towards organic farming. And when we are going to organic farming, there may be these transgenic plants are not classified in organic farming.
So I request you to work in the next 5 processes.
1 minute, sir. Thank you very much. No question.
The next speaker. So now the house is open for the question what we had about the lecture by Dr. Shashi Kumar, Dr. Anjali Anand, Dr. Shivani Nagar, and Dr. Ajit Kumar Shah. Now the house is open for any question answer session from the audience side.
Any questions, please?
So there is a lot of talk about decreasing carbon footprints in particular, right? I think the young minds are sitting over there. So we would like your recommendations to be looked into so that the environmental goals, sustainability goals can be achieved by decreasing this carbon footprint. So things like that. What do you think? Okay. I was just asking you because you talked about a very hard topic of impact of carbon dioxide. My question is that young minds are sitting over here. Any recommendations like that they could, give back to the society by reducing carbon footprints or something like that, so that they are also having an important society in terms of these environmental concerns.
I think that's the nature important, because more we have concrete, more we producing CO2, more refined steel we need now, rust free, so that also adding more carbon footprint to the environment. So like we using all the resources, electricity we using, we should have our houses more like nature friendly so that sufficient light could come. So if you can reduce then the like coal burning, adding like 44% carbon footprint globally. So we can reduce by consuming energy. Even using solar power-based energy, there are also 33% CO2 footprint is there because these panels has to be produced through where the CO2 is emitted. So it's not also CO2 free. So this is the you can understand. I will not anthropologist to much in this area, but thanks for us. So I'm trying to practice this.
Thank you, Dr. Shashi.
Thank you so much. Thank you, our distinguished chairpersons and esteemed speakers. It is now time to felicitate the respected speakers with the memento as a token of our thanks for the appreciation to take out their invaluable time and delivering the talk. May I first request the chairpersons to please felicitate Dr. Ajit Kumar Shastri, sir, Director NVRI, for taking out his time and giving us
We
the
request the organizing secretary also to join us.
please, sir.
Thank you.
Thank you so much. Now I request
दूसरे डॉक्टर के बच्चा पे क्या है ताकि बताना पड़ता है कि दूसरे डॉक्टर से क्या है
thank you so much. Now I request to please deliver the memento to Dr. Shashi Kumar, sir. Thank you so much. May I request Dr. Anshi Anand ma'am from IARI to please come up on the dais and accept the memento. Thank you so much. May I now request Dr. Shivani Nagar ma'am from IARI to please come up on the dais and accept the memento. Thank you so much. Now to express our gratitude to our esteemed chairpersons for their invaluable guidance and contributions. And our request Professor M. Siddharthi sir kindly felicitate our chairperson, Professor Javed Ali sir and Dr. Shashi Kumar.
Now I invite all the speakers, chairperson and moderator for group photograph.
Just a group photo.
अच्छा है ना कम हो जाएगा कम कर रहे हैं ना आजकल कम कर रहे हैं
पीछे मत गिर जाना
Now it's time for a tea break. I would like to request all our guests and participants to join us for tea. We will resume after 15 minutes for technical session 3.
Kumar Mandal. He's actually principal scientist at the ICAR Net Institute of Plant Biotechnology, New Delhi. His research focuses on the molecular mechanisms of salinity, stress tolerance in rice and its wild relatives, aiming to transfer key genes in microRNAs into salinity sensitive rice genotypes. With over more than 60 publications, his work has made significant contributions to plant biotechnology. In recognition to his research, he has been elected as a Fellow of the National Academy of Medical Sciences and has received several prestigious accolades in the field. So he's going to talk on diversifying the primary gene pool of Brassicas, leveraging diploid progenitor species and crop wild relatives through plant gene strategy.
ये कहाँ पे है?
Take the stage.
Hello. Thank you very much, and I would like to convey my thanks to the organizer, Professor Abdin, for inviting me consecutively sixth year or 6 times. So, as some of you know that I generally talk on the salinity because my lab works on the salinity stress. But this time, for the first time, I'm going to talk on a topic for which I have never presented anywhere, and it is a kind of immature work. But I prefer to choose this because that is the way to develop the or mature the work. So if anybody is having any suggestion, please let me know. So today I'm going to talk on discovery of the QTL on the AON related trait, and the rice. Sir, 20 minutes time, no? 15 minutes. Okay, I'll try to quickly go through this. Edible rice, we know that Oryza sativa. Apart from that, there is one more species called Oryza glavarima, that is African rice. So it is used in the Africa, but they are highly stress tolerant. So in my lab, there was a PhD student from Nigeria. So that way this work has been started. My talk will be having sufficient genetics and breeding work. So if anybody is having any question, they may ask in the later on also. So what we did, a little bit about the AON is that it is a protuberant structure on some of the rice or cereals. It is not present in everywhere, and it has some role for stress tolerance upcoming, and also it does photosynthesis in case of wheat. But it is well studied. Some information is there on drought, but not there in salinity. That is why we prefer to work on this. Otherwise, it is an undesirable character for the breeder. They do not prefer because in the processing in the milling and other places, it creates a problem. Well, what is known about the AON is that there are 3 genes known, RIA1, RIA2, RIA3. And out of that, it has been known that from Bharti, the African rice has come AONless after mutation in the RIA3. But in Asiatic rice, Oryza sativa, it has come on the RIA1 and RIA2. But RIA3 is not mutated. So when we started the work 6 years back, we developed 2 kind of mapping population, taking IR29 as a female, glavarima as a male, and then we developed this real line. Also, we developed the backcross line in which the glavarima was used as a male and PB1 as a recurrent female parent. So we developed the recombinant line. We also developed, we took the phenotyping and we developed kind of QTL-seq analysis for this. Here we did the whole genome sequencing of both the parents and as well as the extreme phenotype, which is having 30 plants is having the AON and 30 plants which is AONless to validate the gene. So we discovered the gene from here for AON development, and then we validated in the backcross population and further validated in the QTL-seq. So whenever people do the QTL work, the major emphasis is that phenotyping data has to be very correct and QTL need to be validated before giving to the breeder or for some other purpose. So this is our experimental scheme. You can see that this is the variation in the real population. Length of the AON, you can see some of the lines have a very high length, some of these have an AONless. And important point is that in the length of the AON varies in the structure in the panicle. In the top portion is the lengthiest one, middle portion is having the little lesser, and the lower portion grain is having the less portion. So we prefer to identify gene separately for all the analysis, top, middle, and bottom. So we have phenotyped that, we have find out that there is a variation, and the variation is skews toward the AONless because 0 length is more. This is the number of plant in case of real population. we have done the Pearson correlation and find the correlation for this trait. Similarly, we have scored the bill population, that is backward inbred line, on which the Pusa Basmati one is the one of the parent and Glavarivan is the other. Very interesting is that there are many, both of them are having A1. In case of their progenies at BC to F4 level, you can see the segregation. Some of them are A1-less, some of them are having longer A1, indicating that the gene has been segregated. We took the advantage of this to validate this. Here also we have done the top, middle and bottom and did the Pearson correlation analysis to check whether they are correlating or not. And after that, we have done the GBS, that is genotyping by sequencing. That is the normal for genotyping the entire population. And from there, we have developed the linkage map. From there, we have identified the gene which is responsible for this. And then we have aligned to the some analysis to separate out some marker. And finally, we have developed the gene. So this is the linkage map of the, sorry. This is the linkage map what we develop. But interesting point is that many times we ignore the some of the marker which is present in the linkage map, which is do not follow the Mendelian genetics. That is called the SD marker, segregation distortion marker. Work on segregation distortion marker is very less because many times they are responsible for creating the mutant or switching of the gene. Here we can see, I'll show you one of the gene, how it has been affected. So whenever we develop some kind of genetic analysis, first we identify the segregation distortion marker which do not follow the Mendelian genetics and remove them because they are no use of us. And only we take the marker which is follow the Mendelian genetics. That is called linked marker. So to identify that in the reference genome of the Glavarima, we have taken this marker. They are red in color. And after that, this is a kind of collinearity test which indicate, you can see the chromosome. These are the 12 chromosome of rice. 1, 2, 3, 4. Like that, you can see that 6, 11, and 12. They are mainly broken, indicating that there are a lot of segregating distortion marker are present. When we look into this, we find out that one gene which is known for Asiatic rice, RIA3, on which some of the SD markers are located, indicating that this gene is not working for our case. So what I want to say, in our population, already known genes, RIA1, RIA2, RIA3 is not present there. So then question is that what genes we have. So that is why we advance our work. This table shows that chromosome 1, 6, 11, and 12 is rich with the segregation distortion marker, and which we finally remove them. And here there was a, already I have explained you that what is known in case of AON development, but to test that RIA3, that is the main gene for AON development, in our population, we took the Oryza varti here, leaf, and we find that this gene is expressing high. But in case of our parental population, that is the Glavarima as well as IR29 and their segregating progenies, this gene are not expressing, indicating that RIA3 gene, which is known for AON development, is not there in our population. So there must be some new gene. And we did some analysis and we know the reason that there is some mutation in the promoter region. That's why this gene is not there. And this is the diagram that how we have done this, I'll tell you. And this is the actual linkage map minus the segregation distortion marker. That means here all the marker are linked to the linkage map. So there, to identify the gene, we use several QTL software. These are those who are familiar, for them it is fine. Otherwise, in brief, I'm telling, we did the inclusive composite interval mapping, internal interval mapping. Different software or algorithm are used for the identifying the QTL, which increases the robustness of the work. And through this QTL mapping, we have identified, in our case, the 4 chromosome which is having the AON development. But when we plot this, you can see that chromosome number 3, we initially identified with one software some gene, but not others. So we have rejected this. In QTL chromosome number 4, most of the software has given the gene present here with a phenotypic variation percentage 17.5%, indicating that it has influence for AON development 17%. In chromosome number 8, again, some of the software has shown that they are positive or this is authentic. But chromosome number 12, most of them are shown that it is not having the gene. So all these 4 algorithms, when you use, based on that, you have taken a decision that chromosome number 4 and chromosome number 8 is having the gene. Then what he did, this is the indication of that the same, that the peak indicates the chromosome, that the QTL. So in chromosome number 4 and this is chromosome number 8, you can see the peak. So different algorithm we used, and we find out that this is the two region on which the chromosome is there. There is one more thing in QTL business that is called epistatic interaction, but in our case it is not there. This circle means that there is no epistatic interaction. Same algorithm, another algorithm, they shown that same thing, and there is no epistatic interaction with that. Now this is another, and you can see that sometimes my presentation is written Kharif 2023, Kharif 2024, because in QTL mapping we require minimum 2 years data. So both the years data has been added here. Now in QTL, which is biparental mapping population, people used to this known software to identify the QTL, but for the first time it is coming that we have done the another more analysis called genome-wide association studies. And normally genome GWAS is used for the population genetics, but here in the Mendelian genetics we have used this, which indicates there is a 9 different population that is indicated by this. And then this is the Manhattan plot of the GWAS, which again shows that there is a chromosome number 4, this peak, chromosome number 8, this peak, and chromosome 3 and 12. Sometimes they are coming, sometimes they are not coming. There is no consistency. So 4 and 12 we concentrated for single locus and multi-locus. These are the software through which we can identify. So now we know that chromosome 4 and 12 is having the QTL of our interest, but how do they validate them? Many people, they publish the paper here, but we want to do the details about this. So we did another technique called QTL-seq analysis. Here, both the parental line as well as their progenies, the extreme line, which is having 30 numbers, having a high avon length, 30 is avonless. We have taken the DNA from 30 plus 30 separately, and then we have made a 2 pool, and then 2 pool has been sequenced. So we have the 4 sample sequence, 2 parents, 1 pool of your high avon and one other pool of low avon. And then after that analysis we have shown, so it is called bulk DNA. We have validated this, and we have find out that delta SNP, that is the variation of SNP, exactly falling in the chromosome 4 and chromosome 8, that is absent in 3 and 12, indicating that our bioinformatics analysis was correct. And then in QTL is a chromosomal region which harbors many genes. Now pinpointing the one gene or few gene, we did some analysis, and we have picked up one gene which is known as serine kinase in chromosome 4. And in 8 also, we have picked up 2 genes. One is the box proteins, and another genes are there, which is called SKP1. So why we have chosen this gene? Because gene ontology say they are related to the cell division. What happened in avon? Avon required a lot of cell division to be occurs in the lemma region of the rice seed. So this gene is suspected to be, we find out the chromosome, now we are sorting the gene. So in the chromosome region, there are so many genes, but from there we have picked 1. Here also so many genes, they are picked 2. Now we try to find out there are 2 chromosomal regions, whether there is any relation or not. So far we have identified the QTL, and here one question people ask me that, okay, the gene which you have find out, is there any known gene falling onto this, which is already been discovered for avon development? So we have done the review of literature, and we have find out these are the gene, but our region does not fall with any of the known gene, but close to one gene. In chromosome number 4, there's a RIA1 gene is chromosome number 4. Ours also chromosome number 4. But when you see the map position, we find out that it is nearby, but not exactly in the same map position, indicating that our gene is novel and not reported so far. Now, how to know that it is valid, it is correct gene? So we did the Sanger sequencing of that region for different phenotype, that is avonless, long avon, avonless, like that way we did, and we found that wherever the avonless is there is a T added here. And what has done? And then followed by A and G. So TAG is a stop codon, they prematurely, in avonless gene, they have stopped the protein synthesis. And that is the reason of that our gene is not fully synthesized in case of line on which there is a avonless phenotype. And then we have shown here that what are the region that has been deleted. So it is having the 131 amino acid residue has been stopped in the avonless gene, and that is having the kinase domain. So this gene having the many domains, one of them is the kinase located towards the C terminal, and that is absent in case of avonless. So to further narrow down the genomic region, we have adopted technology called regional linkage map. So there we have taken the SNPs present in that region, and indel present in that region. And we have narrowed down the again further from the 256 kb to 156 kb. So the more you can narrow down the chromosomal region, that is preferred by the breeder, because unnecessarily gene will not be present in that QTL region. So we have done that so far. And then we have checked the in silico analysis of our gene. We have find out that our genes are either expressed anthers or expressed in the root. Here there is a catch, because I told you in the beginning, I work on the salinity. I do not work on the developmental biology. My interest was to know whether AON has anything to do with the salinity or not. So these gene present in the root, and when we check in the literature, we find that similar kind of kinase are act as a signal molecule for the salinity tolerance. Although here it is AON development, but this highly expressed in the root, indicating. Okay. The moment you said 2 minutes, I jump into the end slide. Yeah, I have a 3 more slides. So that way we are suspecting that this has the dual role. Based on the work so far we've done, we have developed a model. In case of IAC29, which is AON-less, these genes are truncated and kinase domains is not there, so they cannot add the phosphorus. Therefore, no phosphorylation down the stream. There is no phosphorylation, and cell cycle proteins, that is your cyclin CDK complex, are not activated, and therefore there is no cell division. That is our hypothesis. Look at the, sorry, this will be the your Oryza glaberrima, long AON. I'm sorry. In the Oryza glaberrima, on the other hand, which is a long AON phenotype, the kinase, this serine threonine kinase in chromosome number 4 are active. Therefore, they do the phosphorylation. They make the cell cycle protein active, and therefore there is a cell cycle, cell division, and this is responsible for the AON formation. And in our study, chromosome number 4, we have got one gene that is called serine threonine kinase receptor. Chromosome number 8, which is F-box protein, and the ubiquinase. So these 3 genes collectively responsible for the AON development or AON-less development. That is our findings. And this is the hypothesis already I explained. So this is the summary. We have developed a cross which took 6 years for all these cross. And after that, originally it was developed for salinity tolerant, but we find luckily that parents are contrast for the AON. And we have identified that HEA3 is not there in our population. We have identified some candidate new gene in chromosome number 4, serine threonine kinase. In chromosome number 8, we have identified 2 genes. And we did the in silico analysis, find that there is a protein in silico followed by Sanger sequencing to confirm that kinase domains is absent. And then we are trying to do some kind of validation phosphorylation to prove our hypothesis. Thank you very much. I acknowledge Dr. Bhumi, who came from Nigeria for doing PhD in my lab, and we introduced the Oryza glaberrima, and my breeder friend, they helped me to develop the mapping population. Professor N.K. Singh is Manter, past and present director. This is my students, and these 3 persons are responsible for this particular work. And this is my whole group. Sometimes, I think 2 years back, the photograph. I don't have the recent photograph of that. Thank you very much.
Thank you for the wonderful talk. So, a chance for a couple of questions from the audience. Light on, please. So, Dr. Wonder, I have a question.
Right, sir.
Before starting the study for the kind of genes that we're dealing with, you are sure that there are single copy genes?
Not really. Kinase is a big family, you know. This it is not a single copy gene, but in our QTL region, when we made it short, so this is the only kinase present in that 150 kb region, and in rice, 10 kb contain 1 gene. So functionally, we have 15 gene. Out of 15 gene, this is there. So that we need to prove. But it is a member of the big gene family, so there could be many other gene also. But in our QTL region, it did not detect. So our validation was based on the QTL what we have identified, and then QTL we have validated in the backcross population as well as with the QTL-seq. So there is a high chance we are quite confident that this is the candidate gene.
So otherwise, I just would like to say it's very commendable. And the whole genome sequencing for hundreds of samples.
Yeah, 200 sample he did.
So you got some good funding?
Yeah, it is a 6 years work. At least 2 funding agency has funded for this. So DST and one more CSR for this. Oh yes. This is the.
Actually, when we do genomic modification or anything, right? Is there any nutritional changes occur between the like RM100 and IR29?
IR29 is a high-yielding variety, very popular in India. It's a very age-old variety. We have taken because it is avonless. And RAM is RAM 100 is a African land race. It is found in the Nile River side. Student brought it. But as long as the nutrition is concerned, we have find out the color of the RAM is little brown. So if it is a brownish pericarp of rice, it is known that they are rich in the iron and zinc. But police rice, when you remove the pericarp, it is white in color. So pericarp may have certain amount of iron and zinc, but since we are not interested, so we did not do this thing. So next is that why I am telling this, because we'll be taking this gene and we'll try to correlate how it is related to the salinity. We know the role of the ion development because my primary interest is to develop the salinity tolerant plant and know the molecular mechanism of the salinity tolerant. And RAM 100 is a highly salinity tolerant. IR29 all over the world used as a susceptible check, means they are highly sensitive to the salinity. So we have chosen the contrast. Fortunately, I also find out that there is a contrast in the AON for these two parents. That is why I'm trying to correlate that does AON has any role for salinity tolerance or not. That is my purpose.
Thank you.
Okay, so now we go with the other speaker.
Okay.
Thank you. Yeah, so the next in the list, Dr. Shomya is on the way. So I'll go with the other one. Is Dr. Yasmin Ahmed. Just to introduce to you, Dr. Yasmin is a senior scientist at the Peptide and the Proteomics Division of DA-PAS and a leading researcher in hypobaric hypoxia and high altitude adaptation. With a PhD in analytical chemistry from AMU, she has identified key protein biomarkers for conditions like HAP and please and advanced proteomics techniques for human and animal models. She has published over 45 research articles. Her contributions have earned her prestigious awards, including Marie Curie Excellence Award and Laboratory Scientist of the Year 2021. Dr. Ahmed also serves on editorial boards and as a reviewer for several publications. So she's going to talk on innovative biotech approaches for high altitude health challenges from diagnostic to therapeutics. Ma'am.
Good morning, everyone. Thank you very much, sir, for the brief introduction. So before I start, I would like to thank Dr. Saima Wajid for giving me this wonderful opportunity to present my work over here. So I have been working in this field of high altitude stress for the last 18 years. And starting with what is it? And so a brief introduction, what is hypobaric hypoxia? So in the next 15 minutes, I will be talking on the topic entitled Innovative Biotech Approaches for High Altitude Health Challenges from Diagnostic to Therapeutic. So what is hypobaric hypoxia? Well, with increasing altitude, there is a non-linear decrease in the barometric pressure, and it causes the generation of the free radical species, and which is responsible for many high altitude related illness. For example, high altitude pulmonary edema, high altitude cerebral edema, and acute mountain sickness. So now the question arises, why we are interested in something that happens in the remote far-flung area? So if we see this this statistic, we will find that 24% of the geographical area is high altitude. Around 140 million of the world populations live at altitude, and approximately 100 million people visit mountains every year. The most important and the critical issue is our strategic deployment of armed forces at high altitudes, which is a very serious concern. So if we look at this map published by Hickott et al. This. Okay. In India, 49% soldiers had acute mountain sickness. This is a massive scale, which affects the army personnel. So it is very important to define the scientific questions, more precisely the rationale of my work. So the key questions are: can we develop the protein signature that helps in the diagnosis for the high altitude related illness? And can we recommend the shortened acclimatization protocol for army personnel? Second, what are the molecular cascades that are affected by high altitude hypobaric hypoxia exposure? Number, can we deliver a side effect free prophylactic interventions for minimizing high altitude related illness? And can we create an animal model that would be help in understanding the high altitude illness in a much better way? So when we are going through the literature, and we have found that there was some crucial lacuna in the literature, that we don't have an early diagnostic marker for high altitude pulmonary edema. We don't know the signaling, what are the signaling pathways. Second, we don't know the acclimatizations markers and what are the post-translational modifications, and no objective based methods to assess acclimatization at high altitude. So now, there was one project in which the aim of that project was to improve the combat efficiency at high altitude. So this combat efficiency means that you have to prepare the army personnel within a shortest possible time and logistical effect. When we are talking about the high altitude, there is a acclimatization. Well, high altitude acclimatization depends on two factors. One is time of exposure, and second is the intensity of the exposure. When the intensity of the exposure we are talking about, that is the altitude variation. These are the two important factors which play a very important role in high altitude acclimatization. Now, what happened? Why there is a and the main problem to achieve this high altitude acclimatization at high altitude is the long period of this acclimatization. So, in this study, what we did, we studied these two factors. First, we study the temporal part, where we have varied, we expose the rat at different time interval. And we have found at that 24 hour of time period, there was a maximum perturbation in proteome. And this is confirmed by the medical literature, which suggests that most of the high altitude illness occurs within the 24 and the 48 hours of the high altitude exposure. So at this study, what we did, we exposed the rat at different altitudes, that is 10,000 feet, 15,000 feet, and 25,000 feet at a fixed exposure of 24 hours. What we have found, that when there was a direct exposure, at 25,000 feet, there was 100% mortality. In order to save the animals, from this mortality, we have given the pre-exposure, that is to expose the rat at 10 hour of high altitude exposure, followed by 1 hour of normobaric exposure. What we have found, that the 100% mortality is converted into 100% survivability. Now the question arises, what happened with the rats, and how this survivability happened? When we perform the proteomic analysis, we identify that at 15,000 feet, when there was no mortality, the rats were suffered from redox stress. Number two, cytoskeletal instability. Third, energy metabolism dysregulation. So what we did, we validated these protein, because there are three things that happen at high altitude and that gets perturbed, three pathways. One is redox homeostasis, second is the energy metabolism, and third is the cytoskeletal proteins. We validated those proteins, and we have found that after this pre-exposure regimen, that is 10 hour of hypobaric hypoxia followed by 1 hour hypobaric hypoxia, we are seeing that these NRF2, PRDX, GPX, and TRX thyroidoxin, these are the antioxidant molecules, they were recovered to the baseline level. So this shows that our pre-exposure regimen is an excellent indicator for the recovery of these proteins. After that, we check the level of cytoskeletal proteins, viminantine, tubulin, and actin. Again, the same thing, the pre-exposure recovery to the baseline levels. Now coming to the energy metabolism related proteins, where we have checked this RXR and MDH and GAPDH. Again, we can see here Same recovery to the baseline levels. We check this lactic and pyruvatic indicator at 15,000. We can see it's very perturbed, but after our pre-exposure, see. Now, this is the MC, pro-inflammatory cytokines at 15,000 feet. It's a stress, and after our exposure, C. And this is a sulfotransferase protein. This is a biomarker for the high altitude pulmonary edema. We have checked in our this. This is not showing any change. It's constant. That means it could be act as a negative control in high altitude related acclimatization. Finally, we have validated the upstream molecules like STAT3. They are downstream effectors, that is calpain-2. Again, we check the levels of these pre-exposure regimen, the recovery to the normoxic levels. And calpain, this shows that there is a reduce in the inflammation after our pre-exposure. Now, this is the overall, we have given a protocol of, 11 hours, and we have seen that these three molecules, STAT3 and RF2 and RXR, this is related to the inflammation, this is related to the antioxidant, this is related to the energy metabolism. These three interplays responsible for reducing the inflammatory response, redox hemostasis recovery, and inactivation of the inflammatory process, reduce milk-folding proteins, integrated alveolar, integrated restore, and finally, the energy hemostasis is achieved. Now, this article is published in Redox Biology. Now, after getting information of rats, we translated our finding on humans. So what in this study, we have taken, the human exposed to two different altitude at 11,000 feet, one army battalion. They were exposed at 11,000 feet, that is at Leh, and second is the Fukche, that is at 14,500 feet. Well, here we can cover both the factors, time of exposure and the altitude of variation. So we validated the proteins which we have previously identified in our multiple hypovaric hypoxia studies. Well, all these proteins were, why we are validating, because they are present in the plasma sample, recurrence in multiple proteomic studies related to hypoxia and high altitude, novelty, statistical feasibility, and ELISA suitability. Now we did the plasma proteome analysis by using the ELISA. So we got some trends. Here are the trends. We can see these baseline, these 3,500 is the first stage of acclimatization at the army has to go through. And second, this is the second stage of acclimatization, that is 14,500 feet. Well, in army, there is some acclimatization protocol that have to follow at high altitude. That is the slow graded ascent. Okay, that is gradual acclimatization at varying altitude in number of days. So this is what we have did. And finally, we can see that TRX is this one is during the first stage acclimatization is showing the decline, and second stage is keep on decline. Second, this TRX is after first day and the 7-day recovery to the baseline levels. This is sulfotransferase molecules, as I already mentioned in the animal study previously, that it is not showing. It's again in the human samples, it is not showing any change. It's the constant. This is a calmodulidine. This is a calcium molecule. It's after 30 days a decline level, and after recovery at 120 days. Again, this GPX. This is not showing any change. Well, this GPX and this TRX, these two, if we see that these are the very, one is decline and other is not decline. That means it is very important to maintain the redox homeostasis at high altitude. So this is a very defining feature of acclimatization. So now this is the paxing molecule, apo molecules. This is a lipid molecule, and this is not showing any changes in the first stage, but some higher levels at the second stage of acclimatization. Same with the HB molecule, haptoglobin, not showing any change. Finally, to check the accuracy of these molecules in population, whether these molecules could be used as the panel to assess acclimatization, what we did the report, ROC, receiver operating characteristics. So in which what we have, if this is the AUC, area under curve value, we have taken the value 0.8, that point, value above itna are usually considered in biological research, considered to be the very good. So we have taken the values above 0.8. And this shows that based on these values, we can say that these following proteins could be used as a panel to assess the acclimatization at high altitude. Okay, sir. Finally, this is article. Now coming, so how to improve acclimatization by using the therapeutic intervention. So what we have taken this silimarine, we explore the potential application of this silimarine by improving its bioavailability and solubility. We did very simple experiment. We sonicated the crude silimarine on water bath, and we have found that there was a reduction in the size from 23 micron to the 5 micron. And this enhances the bioavailability and the solubility of that molecule. Finally, after this characterization, we did the dose optimization. We found that 50 mg per kg body weight is successfully, reducing the ROS and improved. See this ROS after this intervention, we can see the reduction in it, the oxidative MDA and this protein carbonyl. Finally, this is the antioxidant. After giving this silimarine intervention, there is an increase in this. Finally, SOD increased. So, and after all these are the downstream effectors. We also check the hypoxia-related biomarkers. So we check this level of HIF1, VGF, and EPO, and we have found that these are differentially expressed, and they are helpful in reducing the inflammation at high altitude. And after the intervention, is showing the increase. So now this is the profile. This is the inflammatory cytokines. After the interventions, we can see the reduction in this TGF, IL-6, TF. Now this is C. At 20,000, at 25,000 feet, when there is a direct exposure, you can see the inflammation. But after this pre-treatment with this silimarine, we can see the reduction in the inflammation. So now the third is that in order to understand the disease well, we develop an animal model. What we did a very simple experiment. We exposed the rats. To hypobaric hypoxia for 6 and 80 hours. We forced the rats to swim. And the rats were examined using the chest X-ray for the sign of pulmonary edema. We then extracted lungs and blood samples. We reverse translated our previously identified HAPE molecule in this model. We have identified this in the HAPE afflicted rats. There is an increase in the level of this particular protein. That this shows that this is the faithful marker of high altitude pulmonary edema. We want to see whether HAPE is associated with inflammation. We check the level of these pro-inflammatory cytokines. We can see that in this HAPE afflicted, it is rise. So now we also did the histology, and we have found that there was extensive damage and accumulation, suggesting inflammation and leakage of fluid into the lung tissue. So now we conclude that HAPE and inflammation process in tandem and synergistic events. To summarize all that I've presented, one, now we have a rapid acclimatization to altitude. We have a panel to assess acclimatization. We know that these are the side effect-free prophylactics, silimarine that prevents high altitude hypoxia-induced lung damage. We develop a radiologically verified model of HAPE and reverse translated our own biomarker. Now we can say that we know the molecular pathways that are affected by hypobaric hypoxia. So thank you all. These are my research scholars. Mostly all they all are in US doing their postdoc. He's going to wind up very soon.
Okay, so I have a question.
Yes, sir.
In comparison, you pre-expose, and then you do the protein study analysis. Is there any direct correlation between people who are resident population in high altitude and low altitude, head-to-head direct comparison instead of?
Yes, sir, we did that. Yes, there was. Yeah, we did that. We have a publication of high altitude natives also. And actually, sir, there are 3 subject groups. We always one the lower lander. Some is the natives who are adapted there at high altitude, and finally those who are like army, they are going to acclimatize at that particular. So we did the all.
So in the acclimatization scenario, where in animals and humans, what the question that I have is, when is your the time starts? Because they are already there. The soldiers who are work, they are already there.
Sir,
There are 2 days already there. Are animals who are taking them. The moment they land, you start the time. That's the question.
That's the time. For example, sir, in the army, there is a protocol for 644. 6 days at lower altitude, that is at lake, 4 days at higher, because these high altitude is divided into 3 altitudes. Very low, high, and then extreme. And correspond to these zones, these are the stages of acclimatization. Accordingly, the army is going to acclimatize on 644 days. 6 at the low, 4, and then finally at 4. So these are the ones they get.
Actually, when there is a hypoxia condition, or we know that there is a erythropoietin level increases. Now, is there any level of erythropoietin change that we have seen during these experiments? Or
Sir, we have seen that there is an increase in the EPO because the oxygen carrying capacity should be increased. But what happened in the disease case when there is a HAPE, this particular protein get down. So when they are acclimatizing well, so this is showing that EPO is increasing at as the altitude changes. So when there is a the maladaptation, when there is some disease, then this particular protein gets down.
and second question is, the celimerin is actually a hepatoprotective drug. It is well known drug, but it is also helpful for this high altitude.
Yes, sir. Yeah, that's why we have
This is a new finding.
this is in the lung because we extended the application of this particular silimarin to this lung. So this is the one new thing we add.
Thank you.
Okay, thank you.
Now our third speaker, Dr. Soumya right? He is a neuroscientist at Amity Institute of Neuropsychology and Neuroscience, Amity University, Noida, specialized in neuroscience and pluripotent stem cells. He pursued his postdoctoral research at University of Science, Malaysia, where Vellore College of Medicine, where she received Autism Speaks postdoctoral fellowship in translational research in 2012. He published extensively high impact international in the international journals and was part of team awarded the Gandhian Young Technological Innovations Award in 2019 and for groundbreaking research in drug screening against malaria and other. I welcome you, madam.
Good afternoon, everyone. Thank you very much. I really convey my gratitude to the organizers and Professor Abdin for giving me a chance and share my research since last few years with you. It's an honor and it's more nostalgic for me because when as a beginner of my student, I got trained here. So it's really a great thing to be here and present my work. Thank you very much. So today I'll be talking about since last few years my lab has been focused on developing and identifying biomarkers in the field of autism spectrum disorder and other neurological disorder, and mostly I started my journey with ASD because it is a idiopathic neurodevelopmental defects where we with the patients or kids from the clinic come with multiple complexities, and we take this puzzle from the clinic to bench and try to identify the molecules which is relevant to cognition and behavior. So to be simplify this line, cognitions are the neural processes which control our behavior. The kids which come to the clinic, we have to go from their behavior back to cognition and then to cells. So I'll be sharing with some of my some new findings from my lab also, which is mostly based on system biology, in vitro, and other drug discovery applications. To start with, okay. So looking back at the global prevalence, which shows the number will vary, of course, because this is a 2020 article which suggests that 1 in 100 children are found to be autism spectrum disorder. So those who are new to this term, this is a neurodevelopmental defects which comes with multiple behavioral alterations. This can be repeated behavior, this can be social seclusive behavior, this can be intellectual disability, multiple behavioral deficit and repetitive and many aggression as well. So the every kid comes with different kind of neurological phenotypes. And in India, it is recent report. I mean, 3 years back, it was sorry, 1 in 54 has been diagnosed to be ASD. And this is also, again, I say this is a number which has been suggested, but this might vary because many of the kids are still undiagnosed because of multiple reasons, because it is either diagnosed late or they are not diagnosed at all. So as you can see, the complexity or the brain heterogeneity, this can rise because of the epigenetic dysregulation, and this can rise due to the environmental factors, multigenic contributions, either chromosomal aberrations or many mutations, and also many unknown factors. However, if this is a recent article which is published by Eric Cushing's group, which they categorically try to study the life or the life tracking of the changes in the brain from the first trimester to the postnatal stages, and they highlight most of the processes which is involved in the brain development, either in the process of proliferation, neurogenesis. Neurogenesis is a process where newborn neurons are born every day. As just to make you aware of fact, we are having at least 800 to 900 neurons born every day in hippocampus. However, most of them die, very few survive. So this is a crucial process. And cell migration, neurite outgrowth, synaptogenesis, synapse formation, and neural network function. These are the most important factors which contributes to, which contributes to basically developmental stages in the brain, and all these are affected in ASD. So they did a huge genetic study in this, and they found that mostly the neurocortex, the part of the brain which performs all the executive functions, all the functions which is impact our behavior, which shapes our personality and other things, this is hugely affected. So this is also different genomic study was done from the different stages, from as I told, from the first trimester to the postnatal stages, and also the infant stages. All are showing different spatiotemporal changes in different areas of the brain, but major effect is the neurocortex. Now, when we look at the complexity, each child can come with the language, impairment, social deficit, hypersensitivity, anxiety, mood, sleep deficit. You can see how many complexity one kid can come with. So it's very difficult to even plan experiment or expect a neuroscientist to design an experiment looking at the behavior. So when I say cognition, we start looking at the common factors first. That what are the factors, what are the biomarkers that can be common in all of the things. Then we look to the other factors. So to answer this kind of question, so as I told you, my journey started in 2013, 12-13, when I got a award from ASD, Autism Speaks Foundation, for the translation research. I started looking at all the brain cell types, and majorly now I'm looking on these guys, which are called brain macrophages, or the microglia, which are very active or dynamic in the brain. This is the only brain cell in the brain which can actively patrol or do the surveillance of the neural networks. For example, if I'm talking to you, the area of the brain which is mostly involved in the data analysis or presentation or motor activity, you will find this microglia to be there. So usually, if you look at a postmortem brain, you won't find the habitation of these cells, but the area which is more active, the synaptic maintenance of the neural network, this will be done by these guys. So I decided to pursue my questions mostly towards this, because these are very plastic in nature. They not only do the surveillance of the neural networks, they do the immunoregulation, They do the dendritic pruning or trimming of the synapses to make a network. They do the phagocytosis and clearance of the dead brain cells. So to start with, I started working with some of the kids from, this is the one from the Canada, who is having multiple gene dysfunction and all having the major ASD symptom. So then, while pursuing many biomarkers in the last few years, I found, as you know, the brain energy metabolism, one of the crucial one, which is important in the neurodevelopment. We found glucose 6-phosphate dehydrogenase as one of the enzyme, which is, it is involved in pentose phosphate pathway, very crucial in ASD. And we found this is because it is X-linked. Majorly, it is the ratio of boy to or male to female is, it varies because males are more biased. So initially, it was detected as in hemolytic anemia. Nobody knew it has any connection with neurodevelopmental defects, but it was detected when exposed to this component, specifically while taking the fava beans. It was detected as a favism in ADHD kids, where whenever they take the fava beans with G6PD deficiency, they develop the hemolytic anemia. So with this, our lab started looking at how this energy metabolism is going to be impacted in ASD. So as you can see, this is the pathway. It is a normal pathway, which everybody must be knowing in pentose phosphate pathway. G6PD is the major enzyme which contributes, the rate-limiting enzyme which contributes to NADPH. NADPH, it is a cofactor, nicotinamide adenine dinucleotide phosphate hydrogenase, which basically contributes or reducing factor for all the antioxidant or redox biology in the brain. So we wanted to ask these questions, how this axis basically regulates neurodevelopment and how it is impacted in ASD. When we look at the literature, we found WHO has a huge site and they have already categorized the enzymatic activity or mutations in 5. So type 1, type 2. So the kid which I showed, he has basically showing the type 1, either type, I think type 2 category, and it is having severely impacted in the, multiple phenotype in the kid. So if you look at the mutation, there are 400 mutations the WHO has categorized. 186 already are clinically manifested. So we took a charge to understand how this impact the brain, specifically the brain macrophages, which shapes the brain cognition and the behavior. If you look at the ASD, so this kids, the autism spectrum disorder is detected after the age of 2, sometimes at the age of the 4. So there is no biomarker to study. The kids only show the behavioral deficit. So people started looking at the plasma level. So we found the NADPH found to be very low, and glutathione level is also low, and the oxidized glutathione is very high. GSS to GSSG ratio is low, and there are other markers. All this, when we look at all these markers, they are specifically linked to pentose phosphate pathway dysregulation and the rate limiting enzyme. So we decided to look at the different pathways. So majorly G6PD impact the mitochondrial dysfunction, folate metabolism and dysregulation, which is involved in the nucleotide biosynthesis, one of the crucial pathway, and protein oxidation and other lipid peroxidation and other things also. So there are 3 enzymes which produces the reducing factor NADPH, which is called malic acid enzyme, isoscytode dehydrogenase, and G6PD. But unfortunately, G6PD contributes 70% of the NADPH in the brain. So that is the reason people who are born with G6PD deficiency, there are more chances to develop ASD or ADHD. This is the wild type. This is a paper which we published, and in this we showed that the wild type G6PD in human microglia, this can function. You can say detoxification of ROS and folate metabolism, nucleotide biosynthesis, and also neurodevelopmental process. However, if it is mutant, it is not going to do any of these contributions. So we understand that we directly can decipher the function by looking at the mutant which we develop in the lab. So this is human microglia. As I showed you, one of the most plastic and highly active cell in the brain, which shapes our neural functions and behavior. If activated microglia will do all the damages, or if it is aggregated or it is more activated, it will also do a lot of damages. But if it is deficient of G6PD, it will not neutralize ROS. It will not control the immune function. It will not control the neuroinflammatory phenotypes, which is one of the major function. And we suggested that this can be one of the therapeutic target also. So we published this paper in BBA also by proposing the therapy and everything. Then also during COVID, since the microglia, human microglia controls the neuroinflammation, and ASD, kids have a huge inflammatory phenotypes. We decided to look into the neuroinflammation pathways when we found the mutant has, first thing would be having hemolytic signature, and then it has membrane damage because of the lipid peroxidation, and other damages will happen. This is an invited review we published in the COVID because we wanted to check how the deficient glucose 6-phosphate dehydrogenase deficient microglia behaves. So, so this is the, sorry. I'm not very, yeah. So if you look at this kit, this is the two patients we are working. One is from Hyderabad. This has the Kerala Kalan mutation, which is 90% reduction in enzyme activity. And the second one, 60 to 70% with the patient. So this is the two patients we started working. And we found out that G6PD is the only enzyme. It is a brain data, which I'm showing from one or first post-conception week to at least 27 years. And you can see the G6PD is expressed throughout the brain, spatiotemporally. However, the malic and the associated dehydrogenase is absent in the major part of that. So we understood it has a function throughout the brain for microglia and other brain cell types. Yeah. So quickly, we during this phase, we designed a probe because we wanted to see the nitric oxide metabolism, and this probe detected the lysosomal acidification and microglia phagocytotic activity. And this, we designed the COVID RNA, synthesized the COVID RNA in the lab, and we found that when we transfected the COVID RNA, and it can easily, the human microglia, this can induce the COVID RNA can induce the lysosomal acidification, and if it is deficient, with the probe, we actually detected this nitric oxide metabolism, or nitric oxide is localized to lysosome. And then when we made the deficient condition, so it found that the phagolysosome, so you can see here, the conclusion was SARS-CoV-2 protein or the mRNA which we synthesize, it can induce all these functions. But then we designed the deficient condition, and the deficiency impaired the lysosomal acidification. That is the reason the study from USA which showed that people with G6PD deficiency are more prone to COVID and more prone to neurological impact. And we found also that same data which showed that if we restore the NADPH, the acidification also restored. So this data showed because the lysosomal acidification is impaired, the lysosome is no longer able to control or clean the virus from the system. So we designed the CRISPR, we decided the different mutants, which I don't want to show, but this is the summary. We optimized the phagocytotic experiment. SARS-CoV induced the microglia, and then overexpression, we express the clinical mutants in the NO to see if it is impairing NO, and also characterize the mutations. So what we found also in the lysosomal function in G6PD deficient condition would absolutely hamper the NO metabolism and inflammation. And this also we did with the lentivirus. So lentivirus also did the same thing. The deficient microglia couldn't control the virus, couldn't clear the virus. So this we published. So we found that yes, the NADPH label lows in the G6PD deficient, and this will hamper the acidification of the lysosome, and that is the reason it cannot clear the foreign particles in the brain, and it will lead to neuroinflammation. So another therapy quickly, I'm just showing. We also use the KEEP and NRF system for designing the therapeutics, because this is the only system which controls the strongly redox biology or antioxidant status in the brain. And we decided to look whether restoring this axis can also restore the function of the microglia. Yeah. So to conclude here, also we supplemented the oxidative stress, the G6PD deficient microglia with different citric acid and malic acid, which are supplementation. As you can see, it has reduced the ROS to 90%. So the deficient can be restored by the other two molecules. And then we also tried diacol, it is an algal fluorotanine, and resveratrol. These two compounds we tried, and this also showed it inhibited the KEAP, which is an adapter for E3 ligase, which degrades the NRF2. So we found these two molecules actually restore the complete function, or it induces the reduces the ROS, and it restored the antioxidant system intact in the neuroinflammatory condition. So quickly, this is the condition you can see here. We have used diacol, resveratrol, and other supplements. It has reduced the ROS and restored the NADPH level, and it controlled neuroinflammation. So this is also, it has restored the lysosomal acidification in deficient microglia, suggesting if we restore the acidification in lysosomal, we can control the COVID neuroinflammatory phenotype also. So this is also, we recently published in Helion, in which deficient condition was made by or supplemented by diacol resveratrol. At the same time, we restored the redox homeostasis and lysosomal acidification in the brain cell microglia. Thank you very much. And this is the team and some of my PhD students. We work in close connection with Professor Shailja in the field of drug discovery. Thank you very much.
Now, it's open for question and answer. Please. Any questions from the audience? I'm having one question. Can you suggest any preventive nutrition for the mothers for preventing the ASD?
There is no diagnostic parameter available to first to detect at the age of 2. Once the kid is 2, it is already done. So the damage is already done in the brain. So we can only restore the cognition or behavioral by supplementing that or targeting the pathways which is hampering. So there is no way to prevent that.
During pregnancy?
During pregnancy, yeah. So that's what, yes, that's what we have done recently. What we are doing, we are getting the clinical data from the prenatal stages, and we are analyzing the pathways which is dysfunctional, specifically in the energy metabolism, and we have suggested the supplementations because drugs cannot be designing so fast, and that's what we did in one kid actually, but the kid is already there. But for the next, because it is genetic, we suggested to the mother to have this supplementation like resveratrol and other, which we find out in the pathway analysis, to take as a supplementation with dietary supplementation to prevent the thing. But
resveratrol can help.
Absolutely. Even the diacol also can help. In fact, there are 5 factors the WHO has highlighted which is impacting ASD. It's maternal infection, post-conception, it is a father's age, and it is dietary toxins which we take from the food, over-vaccination, and stress, oxidative stress.
That means you can suggest mothers to take
Absolutely. And
grapes,
yeah.
so that they can get more resveratrol and they can prevent the ASD.
Yeah.
Thank you. Thank you. Now our next speaker, Professor Rajendra Singh. Dr. Rajendra is a distinguished professor at ACBR, University of Delhi, and is one of the India's leading enzymologists. His research focused on identifying novel regulators of acetylcholinesterase and carbonic anhydrase, key enzyme linked to dementia, and developing small molecules to correct misfolding defect in crucial brain proteins. Dr. Singh has received numerous accolades, including the Board of Director of Fellowship and outstanding scientific awards with 85 publications in high impact factor journals. He actively contributes as a guest editor and editorial board member in several prestigious scientific journals. I welcome you, sir.
यहाँ से भी कर सकते हैं. Good afternoon, everyone. I would like to thank the organizing committee members for giving me the opportunity to present my talk here. And Regarding my laboratory researches, I'm basically a protein chemist, and I try to study the folding and the aggregation behavior of some of the important proteins that's associated with the neurodegeneration, especially amyloid beta, transthyretin, and the alpha synuclein, etc. And also some of the major enzymes like carbonic anhydrase and the acetylcholinesterase that's being linked with the uh, development of Alzheimer's disease or dementia. So today I'm going to speak about a particular enzyme called acetylcholinesterase and how we may try to modulate its function for the therapeutic purpose that we can use as a novel therapy. So, uh, as we know that when we come to the dementia, the term does not specifically refer to a specific disease, but it's an umbrella term used to describe a set of symptoms that can include changes in memory, reception, reasoning, attention, thinking, judgment, languages, etc. These are that are severe enough to interfere the normal life of the individuals. In fact, there are four types of dementias, depending on what type of toxic species are being associated with it. It could be Alzheimer's disease, Lewy body dementia, frontal dementia, and the vascular dementia, etc. Out of which Alzheimer's disease dementia is the most common type of dementia. When we come to the pathophysiology of the dementia, there are at least two schools of thought. One is the amyloid hypothesis, another is the cholinergic hypothesis. According to the amyloid hypothesis, it is being believed that uh, an increased accumulation of beta amyloid plaques results in the mitochondrial dysfunction that results in the increase in oxidative stress, and eventually the increase in oxidative stress is considered to be responsible for the apoptotic death of the cells, which could be via various multifactorial process. For example, increase in ROS level could induce neuroinflammation and also increase caspases and also increase accumulation of several toxic inclusions like A beta plaques and the neurofibrillary tangles, etc. So, when coming to the cholinergic hypothesis, it states that these changes, decline in the cholinergic system precedes the molecular events associated with the uh, uh, beta amyloid hypothesis. Accordingly, it is believed that there is lower production of acetylcholine from the pre-terminal to the synaptic terminal. As a result, since these acetylcholine are neurotransmitters uh, that's going to bind to the specific cholinergic receptors in the post-terminal, and that helps to initiate the action potential. So since there is lowered production of acetylcholine in the synapse, so these receptors could not be activated, and hence there is development of synaptic dysfunction. But however, there is an existence of an enzyme called acetylcholinesterase that this is responsible for the breakdown of the neurotransmitter acetylcholine back to acetate and the choline. And therefore, if there is a way to prevent or to inhibit this acetylcholinesterase, we will be able to populate the pool of this neurotransmitter in the synapse that will help to initiate the action potential again. As a result, most of the drugs that's been prescribed for the treatment of the dementia as of now, including tacrine, donepezil, rivaroxaban, galantamine, etc., are all acetylcholinesterase inhibitors. So, but however, the use of these molecules or drugs are still challenging because they exhibit large side effects or adverse reaction. For example, many of the individuals, they experiences very low heart rate, hypoxia, and severe convulsions, etc. And almost all of the individuals with the treatment regime, they started developing GI bleeding with peptic ulcers, etc. As a result, many of the individuals, they need to abandon the treatment regime in order to reduce these adverse side reactions. Therefore, there is a need to develop other non-toxic drugs with less side effects. In the brain, there are existence of certain small molecules that plays a very important role and important for the neuronal physiology. And out of which methylamines represents a unique that comprises of betaine, sarcosine, dimethylglycine, and the TMAO represents a unique class. Is they are highly populated in the brain tissues and the CSF, and they also exhibit neuroprotective effects. With regard to their structures, all are derivatives of the glycines, except with the varying degrees of methylation, and except TMAO that has a oxygen radical linked to the nitrogen atom. But importantly, this TMAO has been reported to be a neurotoxin. Its increased level has been found to be associated with the development of large number of neurodegenerative diseases. Importantly, on the other hand, other methylamines that I'm showing here all are considered to be neuroprotective is they are important ligands or agonists for important receptors and the enzymes present in the neuronal cells. And importantly, based on the dementia perspective, all these betaine, DMG, and the sarcosins, their level are drastically reduced. For example, betaine was present around 20 to 70 micromolar in healthy individuals, but it goes down to 10 micromolar. And the DMG, though its concentration is low, around 2 micromolar in the healthy individuals, it goes down to 0.1. The concentration of the... Okay. Okay. So with regard to sarcosin, there are no numerical values reported, but however, decreased level of sarcosin has already been reported in the individuals. Next slide. So we thought that it would be interesting to investigate what these methylamines have affect the structural and functional integrity of the acetylcholinesterases. For that, we took the purified acetylcholinesterase and treat with different concentrations of methylamines. As we see here, these are the raw data, time-dependent enzyme hydrolytic reactions, and these are the calculated percent activities, and we see that in the presence of sarcosin, DMG, in the betaine, there is 40 at the highest concentration of the compounds used. There is 40 to 60% decrease in enzyme activity. We also perform an alternative experiment in which we directly use the we extracted the brain lysates and the extracted the protein and then directly treated sarcosin, DMG, and the betaine in the system, and we still see decrease in the enzyme activity of this. So these results clearly led us to believe that yes, these methylamines could really be able to alter the activity of the enzymes. So we were further interested to look at the rationale of the molecular inside, how it becomes the inhibitor of the enzyme. For that, we thought that it could be related to the decreased protein stability, or it could be related to the, say, for example, because of binding to the native state, it might have induced formation of high order oligomers. So to look for it, we undergo heat-induced denaturation and mechanical denaturation. We found there is not much significant difference in the DM and the delta oxygenate of the indicating that the stability is not a parameter for it. The absence of any increase in the light scattering intensity and the 10 images further indicates the fact that there is complete absence of any possible oligomers in the system. So we also look at the structure of the data. So by employing various spectroscopy tools, and we see that there is not a significant alterations in the structural characteristics of the in the absence and the presence of these compounds in terms of near UVC, tryptophan fluorescence, acrylamide benzene behaviors, and also the AMS binding. So this data further indicates the fact that yes, the structural these compounds does not also largely affect the structural integrity of the enzyme. So if we look up at the catalytic side of the acetylcholinesterase, there are presence of two sides. One is the anionic side and another is the acidic side. Anionic side is responsible to bind to the choline moiety, and the acidic side is responsible to bind to the carboxylate tail of the acetylcholine. And again, if we happen to look up the structures again, there is large similarity in the structure because there is presence of the choline moiety and acidic tail associated with it. That has really led to believe that most probably these compounds might be binding to the active side of the enzyme and inhibits the catalysis. For that, in order to value that further, we undergo industry docking studies of these compounds by using light software, by intentionally making grid and active site residues. And this is the data for the docking score and the light energies. And we found that all these in the presence of methylene except sarcosine, this is for the natural substrate that all this retain and DMT, they are having high alkaline binding energy as well as light store, indicating the fact that these are also appropriately fitting in the same pocket where the natural ligands are being bound. So this again let us think that most probably, since it is binding to the active site of the enzyme, this could be standing as the competitive inhibitor. So, in order to prove that these are competitive inhibitor, the easiest way is just by raising the substrate concentration, because in biochemistry we know that for the competitive inhibitor to happen, there is always a competition between the ligand as well as the substrate. So, if we happen to increase the substrate concentration, there must be a reversal of that inhibitory effect. So for that, this is the normal substrate concentration used, 30 micromolar, and then we double and triple the fact that once this methylamine has been introduced, there is an inhibitory effect in all these cases. But however, upon addition of these, upon increasing the substrate concentration, there is further reversal of in all the cases, suggesting the fact that all these methylamines are really competitive inhibitors and inhibiting by binding to the active site of the enzyme. If we look at finally in the active site of this cholinesterase, it contains an entrance pocket, a gauze, and then the catalytic center. So any molecule that's going to enter in the catalytic center, they have to bind to the entrance pocket, that's called the peripheral enemy site, binding of which allows the opening of the gauze, thereby allowing the substrate to enter, and then catalysis can occur. So that's why it is essential for us again, if all our methylamines, they essentially interact with the peripheral enemy site. It has already been reported that thiaflavin T, that is a fluorescent dye, it's a specific ligand of this peripheral enemy site, and therefore it can conventionally be used to study the effect of any ligands that is binding to these residues. So for that, we intentionally titrated the enzyme with different concentration of THCs, and we got a hyperpolymer. So theoretically, if our ligand is binding to the site, then there must be displacement of the THC, and therefore there must be a decrease in the THC fluorescence. That's what we see that in all these cases when we added sarcosine in the region, there is further decrease in the fluorescent intensity at all the THC concentration. Even the proper saturation could not be reached at the extreme THC concentration used in the study. We also calculated the binding constant of THC, that control was 1.39, and this has gone down to 0.73 to 0.86, that is around 50-fold decrease. Again, these data completely believe us that yes, it is interacting with the peripheral anionic site of the acetylcholinesterase. So our next curiosity is whether this idea can be translated in the cellular system. Is all these acetylcholinesterase inhibitors that's been conventionally used for treatment of dementia has already been reported to reverse the A-beta toxicity because of the fact that since there is a good correlation between the increased acetylcholinesterase neurotransmitter as well as the A-beta toxicity. So for that, we treated the cells with A-beta, and first of all, we measure the cell viability. We see that there is decrease in the level, and we use donoprazole as a control, and there is reversal accelerated sarcosine. And this is a slide that's showing that I don't know, this is missing somewhere. This individual effect, they are not largely, they are not that cytotoxic in the cells. We also look at the apoptotic status by taking acrylic orange and ATVR dye. Acrylic orange, as we know that it's dye that specifically interact with the DNA of the healthy cells, but ATVR on the other hand, it interact with the DNA of the dead or apoptotic cells. We see that upon addition of A-beta And this epithelial gives bright green fluorescence and a bright red fluorescence in this case. With A beta, there is a decrease in the green fluorescence and the increase concomitant increase. But however, donor treatment here is concomitant increase here and decrease in this case. Similarly, except sarcosine, we found that betaine and the DMG treatment also reverses the similar behavior. So we also look at the ROS level. A beta treatment increases and then donor reverses, and these again treatment except sarcosine reverses again. This very affected. So our next curiosity is whether we can also combine, and if it is having a synergistic or it could enhance it immunity effect. So we measure the activity again, and we see that with donoprazole, we use all these drugs and mix with the, and we found that there is a more inhibitory effect in the presence. That's true with the betaine treatment also. We again look at the cell viability, and we found a little increase, and also look at the apoptotic status. With the donor control, there is reversal with the betaine and the DMG treatment, and ROS status also having the same. Since yes, and now since betaine and DMG have already been FDA-approved supplement, this is used for homocystinuria, and the DMG is used as performance enhancement. Keeping in mind that their levels have been lowered in dementia, we suggest that they can be used as a part of the supplement, and also appropriate strategy to populate DMG would be an interesting strategy. Again, we also discovered that this also increases the inhibitory effect of this, and therefore this will have to lower their doses and also the lower side effects, and therefore can be used as an adjuvant therapy for the same level. Thank you very much.
Thank you, sir. Okay. Now it is open for question and answer. Any question from audience? Please.
Yes, please. In Alzheimer, most of them are symptomatic treatment, as I said, from tetrin to galantamine. But there are certain drugs that will FDA approve, some antibodies. But these antibodies, their side effects are too large, and therefore patients they do not want to take. And therefore, people are completely relying on the symptomatic treatment only by taking these acetylcholinesterase inhibitors only. Yes, please.
So, what did you mention? Different drugs. So, they have a acute effect or symptomatic effect?
So we, in fact, it was having a more pronounced effect as compared to the tetrin, galantamine, rivastimine itself. But I would say that it is a kind of synergistic effect. It's not an additive effect. When we quantify the magnitude of the inhibitory effect, it is not additive. But that really brings the fact that yes, in the presence of these, say for example, in the patients we allow to take the supplements, DMT and betaine, and go for the treatment regime with the rivaroxaban or galantamine, that will very improve and lowering the adverse reactions to all. That's the concept. Thank you.
I'm having one question, sir. So when we test any drug for acetylcholinesterase inhibitor, the drug need to pass blood-brain barrier, or if it is not able to cross the blood-brain barrier, will it be very effective or not? Cross the blood-brain barrier.
DMT, they can easily, because I actually slide, they can populate the CSF as well. So at the same time, they are synthesized in the cell also. So we can play, we will try to manipulate the concentration in the cell as well as some over dietary also.
Okay. So betaine is there in beetroot, wheat. It is a active content of the wheat.
Not only that, I would mention that many of the meat consumers, these are rich in choline diet. So betaine basically obtained from the choline intake, and therefore people who consume meat
They have more of the choline-rich diet you suggest for preventing this deficiency. Thank you, sir. Our next speaker, Dr. Sachin Tuekia. Right, Dr. Sachin is a professor in Life Science Department at J.C. Bose University of Science and Technology, Faridabad, specialized in plant biotechnology, gene regulation, and small RNA. He published extensively in high-impact journals and received several awards, including that scholarship travel grant from American Society of Plant Biologists. He he has extensive training and experience working in the field of plant development, stress biology using molecular and biochemical tools, including miRNA and artificial miRNAs. Welcome, sir.
Thank you, sir. Thank you for invitation, the organizers. I thank you, everyone. And so my talk is on artificial microRNAs. So I just want to tell you what the microRNAs are first, and then we will discuss what are the artificial microRNAs. So this is the microRNA biogenesis pathway. That is in animals, this is in plant. As you all know that there is a microRNA gene. The primary microRNA is formed and which is processed by DROSA to form a pre-microRNA, which is further processed and outside, it goes outside the nucleus by exportin and further processed by dicer to form a mature microRNA. So there are two microRNA strands, guide strand and the passenger strand. And the guide strand is loaded on the RISC RNA-induced silencing complex and Argonaute-based complex and induces gene silencing based on translational repression. Similar pathway exists in plants, except that there is no DROSA, but only the dicer, which is doing all the functions of converting primary to precursor and precursor to mature microRNA. So if you are not knowing, microRNAs are very small, around 21 to 24 nucleotide non-coding RNAs, small non-coding RNAs, which are huge implications in gene regulation. So mostly the gene repression takes place by cleavage in case of plants and translational repression in case of animals. So based on these microRNAs, we came up with the design of artificial microRNAs, which already existed, first designed by Wiegel's lab, and we came with certain modifications. What we did was, we found that we kept this is the guide strand, which is followed by a spacer, and this is the artificial microRNA guide strand, this is the passenger strand, and we kept the mismatch at position 1 and 12. Why we kept these two mismatches, we'll come to know. And this design was very highly specific for inducing gene silencing and very highly effective for inducing gene silencing. So this is what, and we also find that if we take a tandem repeat of these two artificial microRNAs, so we can, like two hits at guide and target one gene, they're more effective than one hit. And also you can target several genes together. I will show you with this design. So this is artificial microRNA 1, artificial microRNA 2, all in the same construct, and you can induce better gene silencing. So this is again one hit inducing artificial microRNA binding to the target region 1. Here the two hits, a tandem, two artificial microRNAs arranged tandemly, and we used microRNA 168-1A as a backbone, as a promoter, the backbone of this microRNA or Rhabdopsis microRNA as a backbone to design these artificial microRNAs. Okay, and these are targeting two different regions, and they induce more effective gene silencing. So how we came up with the design? We saw the microRNA, siRNA design rules in humans and Rhabdopsis. You can see that these are the microRNAs found in the humans. And for every position 1, suppose there is position 1, there is A, there is a U, there is G and C. We found that whenever there is A, there is more likely chances of having U, definitely. Similarly, if there is A, again U, these are for position number A. So position number, if there is A in position of 1, 2, 3 to 19, 20. Again, U at position 1 to 20. We found that whatever is the highest matching at each position, we came up with the statistical rule. MicroRNAs found in the human, all the microRNAs found in the database and also in Arabidopsis. Right, same way. Like A mostly pairs with U and very less with A. So these are the statistical things we took into consideration, and we found that there's a mismatch at position number 1 and 12, 1 and 12, 1 and 12. Likewise, these are in plant species. So percentage of positional mismatch, positional base 1 before the seed region and base 12 after the seed region. This was the most highly mismatched region. Here also same thing. This is among the animal system, Drosophila, humans, mouse, and likewise. So we came up with a very static design that we are having a mismatch at position 1 and 12 and designing this microRNA construct. And this is also because, and the microRNA guide strand was very perfectly matching with the target region, which also confirms with the earlier reports of Kai et al. And Park et al. That perfect complementarity to the target is much better for inducing gene silencing. And this mismatch is enhancing the asymmetry and induces better gene silencing. So we were first targeted the GFP. With this construct, we produced 2 microRNAs, 1 and 2, and this is the GFP producing plant, and we found that under 35S promoter, very strong gene silencing with either 1 hit or 2 hit, doesn't matter. So this is the artificial microRNA 1, 2, both targeting GFP, and there was no difference. So 1 hit, 2 single hits, and a double hit, no difference in. But when we used a weaker promoter like inducible promoter, beta estradiol inducible promoter, we found that 2 hit is better than 1 hit, and we also found this is the relative expression of the microRNAs. The same thing we did with the PDS, a gene responsible for this pigment, and we found that again 2 double artificial microRNAs are better in silencing than the single artificial microRNA. And we confirmed many other gene silencing with using this. This is the genetic mutant of CTR1. This is a genetic mutant, and the mutant artificial microRNA resembles with the genetic mutant and shows the efficacy of this technique. Not only that, this is the AGO1 being transformed and silenced using this. These are all the variable phenotypes of AGO1. This is again AGO7, and we see that the targets of AGO7 are upregulated when AGO7 is downregulated. This shows that AGO7 is downregulated by using artificial microRNAs against AGO7. Now we silence multiple ARF genes, ARF7, ARF19 in Arabidopsis, and we can see that this is artificial microRNAs here, and we found that multiple ARF genes can be silenced using this. This also use 5 synates, 3 SNRK2 genes also were silenced. Multiple genes can be silenced if you use a common region which is targeting the target genes. So if you take a common region or a region common in all the multiple genes, you can silence all of them together. If you want to silence one gene out of the multiple family genes, then you can have to target a unique region. Okay, so that's a concept. Here, like all these ARF genes, ARF9, 13, 12, 14, all these were silenced together using a common target region. Okay, so this is the multiple silencing was a Another thing which we used for this tool was not only for gene silencing, but to overexpress any endogenous microRNA. You can use this same tool for silencing as well as overexpressing any microRNA. So we use this, we overexpress microRNA 166. You can see this is the wild type, and we overexpress this microRNA 166 in rice. And you see that the northern plot here showing the overexpression. This is the control. So you can also overexpress any microRNA using this technique. So this is a dual, like serving two purposes, silencing as well as overexpression, both using the same construct. So this is again, this is artificial microRNA targeting 169. No, not the 169. This is overexpression of 169. And we found that the target of 169 is NF by A. So when we overexpress microRNA 169 using this tool, the target goes down. Okay. This is because of the repression, the cleavage. This is in tomato. So these are representative phenotypes. We use artificial microRNA target leafy gene, YABI gene, and tri gene, which induces trichome formation. And we found this interesting phenotype. What we did, we compare our design with the earlier designs of Beagle's lab, like leafy 1, leafy 2, or Beagle design. This is for this lab, for our lab, and this is negative control because this was not silencing anything. Percentage of plants showing phenotypes here, and we found that in one of the construct, like here, this design is better. Our design is better. One of the design in Beagle's lab was better than our design, but most of the cases our design was better. We also put another U in front of, because most of the artificial microRNA is having this A to it with YABI one with extra U. This extra U or extra T was added to artificial microRNA targeting YABI and trigenes, and we found a little bit difference you compare with this, but there was no difference in the other construct, so it cannot be said uniformly. We also look for off-target effects, and we found that it's very, very specific. At some places, for example, this is leafy is down, but other off-target, we looked the off-target predicted by the psRNA software, and we found that they are not being targeted by this. Very, very specific. Some cases there it is like there it is there, and it can be an indirect effect also because leafy is a transcription factor controlling many downstream genes. Same way here, this is tri-typic, and another this is caprice microRNA, artificial microRNA targeting these genes, and they both are down, others are not affected. So this is for the functional genomics of microRNA, to find out the functional genomics and also to find the regulatory networks of microRNA. We have to identify this vector is very, very important. So this is like a Swiss Army knife. You can clone many artificial microRNAs like this and target multiple genes and achieve your functional genomic studies. So I would say that even in the CRISPR era, this technology is relevant because many a times CRISPR can induce lethality. So whenever the knockout is not desirable, you can have knockdown. And so for knockdown studies, all these RNAi and artificial microRNAs are important.
Sir, 2 minutes.
Yeah, I'm done mostly. So this is also the microRNA designer we came up, and if you put any target region here, and you can select like the backbone which you want. We selected the backbone of microRNA 168. It will come up with the designer. You can see the blast to see the similarity. If it is any off-target effect here, then the microRNA designer will make the antisense, and it will make the artificial microRNA structure, and it also forms the primer. So everything you can do with this designer too. And this is the website. This is the preview of the microRNA designer, and you can use any backbone you want. So together, this artificial microRNA tool complements the STTM tool, which is to downregulate microRNA. This is to overexpress any microRNA as well as downregulate any gene. And together with the CRISPR, this is like a holistic approach of knockdown or knockout of genes as well as microRNAs for functional genomic studies. This paper was published in Biotechnology Journal recently, and this is our team, Professor Guilin Tang, in whose lab it was designed, and our Chinese collaborators. And thank you.
Any question from audience?
Thank you.
Question that when you, this silencing and overexpression, is it going to be simultaneously or it is conditional?
No, silencing, when we silence, we take the region of the target gene. Okay.
Yes, sir.
When we silence any target gene, we have to take the region of that gene and put it as a replacing the microRNA. When we overexpress, we take the native microRNA structure as it is and overexpress.
So we can.
You are saying that you can maybe you can do simultaneously also. We have not done. Maybe one construct doing artificial microRNA silencing and one construct.
Yeah, because why I'm saying many times if you target multiple genes, so it will be more complexity in the cellular phenotype or any.
Yeah.
So if you want to control that phenotype for a certain period of time to do the genetic screening. So can we do the conditional one and we stop that and then we induce the silencing part again?
You can do that, and for that we use indicible promoter.
Okay.
So you can use indicible promoter only for that part and then stop the promoter and expression of this artificial microRNA.
Yes, sir. And sir, have you your system has been tried in cancer or any other systems?
No, I think the other tests showed the STTM they showed in mammalian cells. The STTM which is for silencing the microRNA and it worked in the mammalian system. But the artificial microRNA we have not yet tested in the mammalian system.
Thank you, sir.
So nice talk. For such a longer oligo synthesize, you want to target 10 or more genes simultaneously. How do you synthesize such a huge?
It's only 96 base pair. This entire thing is only 96 base pairs, so you can artificially synthesize, or we have a vector. You can just put the 21 nucleotide directly in the cloning site and do it by cloning. Or you can directly synthesize the 96 base pair and directly do the cloning. Either way it will work. So cloning is not a problem. We have a PO2T vector which is specifically designed to clone these kind of things for STTM as well as this.
Okay. So for example, for the one gene you mentioned leafy gene, and you can verify that there is no off-target interaction. But if you are going for many targets simultaneously, then the verification may be too tough.
Verification is tough, especially sir, when we are down-regulating a transcription factor, because transcription factors are controlling the cascade, the signaling cascade. One gene controlling two and two controlling various. So that's why it makes a difference. It's difficult to analyze.
In those cases,
we only look for the similar structures like predictive targets. And look only for those. That's the only thing we can do.
So you demonstrated all of them with GFP.
GFP was no. We
to start with the GFP.
GFP we did to prove that two hit is better than one hit.
Right.
Yeah. So the constitutive strong promoter there was no difference. That was it. Other thing we did under strong promoter.
Strong promoter. Thank you.
Thank you.
So we conclude this session. I thank the moderator.
First of all, I want to thank all the speakers. It was a really nice session. And as a token of memory, we wanna I wanna request first of all Professor Selva sir to come up on the stage and offer the memento to Dr. T.K. Mandal, the first speaker of the day. Now I want to request Dr. Humaira Farooqui ma'am to come up on the stage and offer a token, felicitate our second speaker, Yasmin Ahmed, Dr. Yasmin Ahmed, please. And please hold. Now I want to request Dr. Soumya Pati to come up on the stage and have the memento. I want to request Professor Rajendra Singh, sir, to come up on the stage, and I request Panda, sir, to come to offer the memento to for his like eminent talk. Like it was like really nice talk, sir. And now I request Professor Sachin, sir, to come up on the stage and receive his memento. So please be here. And I request Professor Selva to come in the center of the stage. Now I request Professor Abdin, sir, to come up on the stage and offer a memento for both of our chairpersons.
I request our esteemed chairpersons to kindly felicitate Dr. Imran sir for moderating this session. I request everyone.
I request all the speakers to come up on the stage and have a photograph as a memory for us.
Now I am delighted to invite our guests for the lunch break. Participants, please be seated. You will be leaving after 2-3 minutes. After the guests. Now I request all the participants to have lunch. Students, please, just 5 more minutes. The lunch is coming here. Just 5 more minutes. Now you can talk as much as you want. And we will be resuming after 30 minutes. Just 5 minutes.
I request everyone to settle down, please. I hope the lunch would have satisfied you all. A very good afternoon, everyone. I hope you all are re-energized now. We have some engaging talks lined up. So here we begin our plenary session 3. I would like to invite Dr. Javed Ahmed, Assistant Professor, Department of Biotechnology, Jamia Hamdard, to moderate this session.
Thank you, Adit. Good afternoon, respected guests, speakers, and participants. Welcome to this plenary session. It is my honor to introduce our distinguished chairperson, Dr. Humaira Farooqui ma'am, and Professor Zeenat Iqbal ma'am. Please join us, dais. Welcome, ma'am. May I request the chairperson to introduce our today's speakers and continue the session.
Bismillahirrahmanirrahim. Assalamualaikum. I trust I'm audible. Thank you so much. So, needless to mention, it's a privilege to be here, and that too as a chair, I must profusely thank the organizers for giving this opportunity. I belong to School of Pharmacy, so this is again a learning session for me, and I'm extremely happy to introduce the first speaker, Dr. Amulya Pandasa. May I request you, sir, to please come onto the stage. A round of applause. It said, humility, thy name is Amulya. So we say that when it comes to being humble and being full of humility, it is Dr. Amulya Panda. And the man who stands in front of you is none other than the former director of the National Institute of Immunology. He has been the mover and shaker as far as biotechnology and your pharmaceutical industry concerns. As of now, he is the associate director at Panasia. So his contribution is immense, and I have to put on record that he has hugely contributed to our school and particularly to our group as well as a researcher. Thank you so much for being here, and I'm extremely happy that he is going to talk about something which is very interesting to all the youngsters here, and that is translating innovation in biotechnology. We welcome you, sir. Thank you so much.
Okay. ये बंद कर दो थोड़ा. Thank you, Professor Jinat. Thank you, Professor Abdin. It is always a pleasure to come here. Okay, before that, I told I'm going to tell you a very simple term, and this lecture is only for students. If you see, I have written 3 terms: biotechnology, innovation, and translation. Everybody talks about that, right? Everybody talks about biotechnology. Everybody talk about innovation nowadays. Even the politicians are talking innovation. Everybody talk about translation. Whatever you do, everybody is asking, are you doing basic science or are you doing translation science? I still don't understand. But in last half an hour, I'll be able to convince you and define you these 3 terms. I'm sure all of you know, but I will try to give an explanation the way I think what is biotechnology, what is innovation, and what is translation. And I'll give an example. I don't think I'll give enough data or things like that, but I think because you are all students, you should be clear about these terms and terminology. Otherwise, you get lost. You know, it's like if you go towards Chandigarh, people make lassi in the washing machine, and they say this is innovation. In fact, that is jugaad. This is simply extension of centrifugal force of physics, nothing else. If somebody thinks running lassi in a washing machine is an innovation, only God can help him, nothing else. So, being science students, you should have absolute clarity about what is technology, what is innovation, and what is translation. Before I go, let me tell, because I work in Panacea Biotech. It is around 3 kilometers away from this. I don't know how many of you know about Panacea Biotech because I get my salary from there. It is nice to tell about this. We have been making vaccine for last 30 years. I'm sure during COVID, everybody know what a vaccine is. You are all survived because you have taken vaccine. Vaccine is a poor man's drug, and it is generally said if you don't breathe, you will live maybe for 2 minutes. If you don't eat or drink water, you will live for a month or two. And if you don't take vaccine, you will live for a season. You all are alive and healthy because in the childhood you have taken more than 7 or 13 vaccine, DT, TT, polio, rubella, measles, and they have protected you. So that's what we do. We do many vaccine, but we take credit of making this country polio free. You must have heard our country is polio free. This was a mission by the government of India almost 30 years back. But we are the company who supplied 10 billion doses of polio vaccine. And you must have all taken that polio vaccine. And I am sure you all are hale and hearty because you took and survived and you are happy. So this is what Panacea does. Then I'll come back to the outline. I'll tell about the terms and terminology. I think, as I told you, I'll tell you. Maybe this is very preliminary, how to explain biotech or how to do translation because most of you are students. I'll put my way of definition. Then I'll talk about translation. What is that? Everybody thinks that you should do a translation science, not only in a university, in industry, as well as in government. There is no need to do translation always. There must be some blue sky research. Not everybody has to be a Thomas Alva Edison. Somebody has to be Neil Bohr or Rutherford so that you understand the basics, beauty of science. Without that, you cannot do. Then I'll give you an example how actually a idea or a problem can be used to do translation. I'll give the example of recombinant insulin. I don't know many of you know recombinant insulin is the product which is the first product of modern biotechnology. It was cloned and expressed in 1978 and 9 and 81 it was in the market. So we'll give that example. So let's see what is the terms and terminology. You all define. This is the actual official term. It was coined in 1984, technological exploitation of biological process. When you say biological process, it can be plant, it can be animal, it can be insect, it can be virus. Basically, you take a component of a biology, it can be an enzyme also, or a part of the body. And fortunately, it was coined in the 7th IBS Symposium in 1984 in IIT Delhi. I just graduated that year and is nothing but biology and technology. It's not biology plus some ultracentrifuge or flow cytometry or confocal microscopy. It is the technology. The technology means it is science, engineering, mathematics, and economics. When you say it is technology, it is deliverable to the society. Without that, it is just a technique or a lead or things. It should be very, very clear about what is this technology all about. In fact, biology, as you know, has four molecules. You all know protein, nucleic acid, carbohydrate. And because proteins are more active in small amount or specific, that's why most proteins are drugs. In fact, I have another way of explaining what is biotechnology. For me, biotechnology, because I have an engineering background, for me, biotechnology is nothing but mathematics. What you do in mathematics, it's very simple. You do four things in mathematics: addition, multiplication, subtraction, and division, right? Forget about the complexity. Only four things you manipulate. All you do in biotech is manipulate four things: lipids, proteins, nucleic acid, and carbohydrates. I think you all know. nucleic acid replicates, like it is multiplied, carbohydrate degrades, nucleic acids, proteins are added. So it is nothing but mathematics. So don't get scared of biotech or mathematics. It's simple rule. The way mathematics is simple, all you do is play around with addition, multiplication, subtraction, division. In biotech, you just play around with nucleic acid, protein, lipids, and carbohydrates. Come to innovation. This is in what everybody get confused. You must have studied this in the school. Columbus discovered America. You discover things which already exist. I'm sorry, I'm going back to the school, but that is what is needed. Galileo invented telescope because it doesn't exist. Look, Edison innovated electricity. The difference between invention and innovation is, for invention, you spend money. Like here, Professor Abdin or Professor Tripathi or anybody here invent because the government puts money. For invention, you put money, you invest money, whereas for innovation gives you money back. So you can tell, like Galileo, like Edison innovated electricity. I'm sure you all know how much money the electricity generates in the whole world. Same way. So you can say money gives knowledge. That's invention, because we understand things, you do research experiment, you find new things, you patent, then you invent. And when that knowledge gives you money, that is innovation. So you can say innovation is reverse of invention. So don't get confused. This is invention, this is innovation. Innovation is always associated with tangible benefits for the society. You may develop a small sequence or a kit or a DNA sequence. Till that is utilized by the people at large, it is simply an invention, it is not an innovation. Now, let's see what is translation. Very widely used. Everybody asks, do you do translation? Let's see the world. You know, you all know nucleic acid to protein is translation, right? Very big, no big deal. We all know this. But dive deep into this word. What happens when nucleic acid go to protein? Nucleic acid, proteins generally work on conformation. Nucleic acid has conformation, but nucleic acid has sequence. So when nucleic acid goes to protein, there is a physical change, because protein predominantly work on conformation. Of course, the nucleic acid go to protein, there is amino acid, there is a chemical change. So translation means, in this biology concept, there is a chemical change and there is a physical change. Chemical change is peptide bond, physical change is the conformation. And you must have studied in school, chemical changes are permanent changes, like disulfide bond or a covalent bond, whereas physical changes are visible changes. So only visible changes happening in the society can be called translation. That's why I tell, you can say I am doing translation when you get permanent visible change in the society. That means when your discovery reaches the people who need, whether it is in terms of an environmental solution or a new variety of plants or a new vaccine or a new drug, unless it is utilizable, unless it sees the light of the day, actually in the market, you cannot call it a translation. It is very difficult to differentiate, and many people, even including I also face the same problem. Are you doing basic science or a translation science? I keep saying there is no need. As I told you earlier, there is no need that all of us should do always translation science. No. It's like there is a difference between Rutherford and Niels Bohr and Louis Pasteur and Alva Edison. Louis Pasteur developed vaccine for the people and money. Edison innovated electricity only for money. But look at Rutherford and Niels Bohr. They are only interested in understanding how the atomic structure works. We need people to understand the intricacy of the universe in terms of physics, chemistry, math, and biology. We need that. If you are interested, do translation. It doesn't mean everybody should do translation. It's like in a society, you need different type of people. Not everybody will make a kirana ka store and then start selling. You need people, you need different varieties of people like that. And then how do then define what is translation? I, after going through many way, I have made a simple rule. apply this. If it fulfills, then you are translating. If not, do the basic science. So let's see what is the rules of translation. These are 5 rules. I'll tell you, like you have Lipinski 5. I say this is 5 rules. Is it useful? Science is always useful, but maybe not immediately useful. So, like you want to understand why the sky is blue, or does plant know where the roots goes? These are the fundamental questions. Or know how does the human develop in a developmental science? These are the fundamental science. Doesn't need that it will need it. The information may be needed, but the first requirement is it needed or it is immediately useful? Second, is it reproducible? Let me tell you, all translation is not only application oriented. It is based on very good fundamental science. It's like if your seeds are strong, your fruits will be okay. It's like that. So the second question is the science reproducible? The third is, have you find the proof of principle in relevant model? This is very, very important in a healthcare system. Like you want to cure diabetics, or you want to cure malaria, or you want to cure TB. You may do an experiment in the lab, maybe in a cell line or in a mice model, but that doesn't represent the disease. Please remember, disease is human, takes place only at a tissue level, not at cell culture level. In cell culture level, you may grow some cancer cell, put a anti-cancer drug, or put a compound which will kill a TB bug, but that's not the disease. Disease happen in a pathological condition, in a tissue way. Whereas in the lab, you may have a cell culture, or you may maximum have a small animal model. That doesn't represent the human. You see, if you see many cancer drugs, we have used it in animals, mice, all are clear. But the moment it goes to monkeys or human, it fails. So the real model is important. And fourth, is it scalable? You may develop something, but if it is not available to large in public, there is no use. I'll give one example. I am sure all of you must have studied in school while making oxygen. Remember your 6th class or 8th class? You take potassium chloride, add manganese dioxide, heat it, you'll get oxygen gas and collect it by water displacement, right? This is what everybody has read, how to make oxygen. Nice experiment is explained. Have you ever seen this method to be used in plants? No. Of course, that is one of our lacuna of our academic or educational curriculum. What is taught is not experienced at all. Nobody in the world made oxygen by heating potassium chloride and manganese dioxide. In the lab, the experiment is very clean. You take in a tube potassium chloride, grind some manganese dioxide like a catalyst, heat it, and small amount of water is there. Then you have oxygen. You collect it by displacement. Think you want to make 1 ton of manganese oxygen. How much of manganese dioxide you need? You cannot throw it. You need to reuse. Actually, oxygen is purified by liquefying the air. Air is plentifully available. It has 21% oxygen. If you want to make oxygen, just fractionate and liquefy. That's how oxygen is made and available in the cylinder. So scalability. And number last, is it sustainable? I think this program is all about sustainability. You may develop a technology, but will it work forever? I'll give an example. Like if you see last 20-25 years, we have been trying to make lignocellulosic biomass to ethanol. You have wonderful people, isolate organism which will hydrolyze the lignocellulose to glucose, then ferment to glucose to ethanol, then ethanol, and ethanol is used for blending. That's a wonderful way of doing science, but is it sustainable? The question comes. Please remember, we are making around 200 million liters of milk every year because of our veterinary population. We plan, because we have 1.6 billion people, we plan to get 300 million liters of milk in 2037. If you want to make extra 100 million liters of milk every year, you need more cows and buffalo to eat these residues. Where you will get the residues? If you don't have residues, then how do you make it now? So this way, you have to see anything you do, should be sustainable. So 5 points. If at all you are doing research, ask, is it useful? Is it based on wonderful science? Is it reproducible and appropriate model? Is it scalable? Is it sustainable? It looks very fantasy way, but I'll give one example to prove how these 5 points really work. So any science which fulfill the above 5 points are ready for translation. Let's see how to fulfill this. I say this 5 point rule of five, like Lipinski rule. I'm sure anybody who has studied long back, Lipinski developed a normal rule for discovering drug, maybe 50, 60 years back. So any molecule will be a drug. It's low molecular weight, it's log 5 hydrophobicity, it has less than 5 hydrogen bond, it has less than 10 hydrogen. Actually, these are multiple of 5. So there is a Lipinski rule of 5. All I want, you should have these 5 rules. Is it useful? Is it reproducible? Has it proof of principle has been very, very strong? Is it scalable and is it sustainable? Let's see. I'll give an example how these 5 points can be done. I come back now to my area where I manufacture. For me, manufacturing nothing but use the biology, whether it's a plant cell, animal cell, insect cell, or anything, as a catalyst to get a product. And if you look at the product, I call these are the 7 wonders of the world. Look at these 7 things: penicillin, glucose syrup, insulin, serotonin, hepatitis, antibody, and recently the CAR T cell therapy. These 7 products of biotechnology has been used for last almost 100 years and has produced billions of dollars. Penicillin, insulin, erythropoietin, all these things, and card therapy. So anybody wants to do a translation or want to do a technology, I'll suggest pick up any one of them. One is from fungus, one is from bacteria, one is from E. coli, one is from plant cell, one is yeast, one is animal cell line, and this is human cell line. You can take this example, and these are proven example of industrial biotechnology where science technology has developed, used, like we all use penicillin, we all use insulin, we all use hepatitis B, and we have started using cancer therapy. I'll just give one example of insulin, because insulin is one molecule which revolutionized the whole recombinant thing. In fact, if you look back, in our country, we started biotechnology only after the insulin was marketed. In fact, Department of Biotechnology started as Biotechnology Board in 82. Then it became Department of Biotechnology in DST. Then it became independent. So let's see insulin. We are the diabetic capital of the world. I'm sure many of you who do molecular biology, you clone express insulin, nanogram, milligram, microgram, like that. In a gel, you see, you feel happy. But look at the requirement. 500 million, we have 300 million people. If 10% is type 1 diabetes, each one need 500 milligram. So 10 million into 500 milligram is 5 ton we need. 5 ton, 5,000 kilos of insulin. Can you make it? We have been trying for last 40 years. We make around 800 kgs, but we still lack 4.2 tons. So this is the need, right? The first principle. This is the need, and we need to produce it. We have been trying, unfortunately, but I'll give an example. How do you make this insulin? If you're a molecular biologist, you can clone in E. coli, you can clone in Pichia, you can clone in CHO. Because not only you have to make insulin, you have to make insulin in a cheapest way. 500 milligram can be made in $10, $20 because people should afford. Look at E. coli, it produced in 10, 20 grams, but E. coli is very fast, double in 20 minutes, maybe in 1 day cycle. Is many a company in India, because E. coli is very complex, inclusion body, nobody knows this. They make Pichia, it makes good amount, but it takes 1 week. The time is money. And same thing with CHO. So we pick up this E. coli. You can express 10, 20 gram per liter in 1 day batch, and you can produce for the ton. But the problem with E. coli is the moment you express, it forms inclusion bodies. Inclusion bodies are denatured proteins, very, very difficult to renature. What you do is you need elaborate solubilization refolding process, then recover the protein. But in spite of that, most of the commercial proteins are inclusion bodies. So the challenge here is This is how proteins are purified from inclusion body. It is inside the cell. You take the cell, lyse it by French press. You have denatured protein, solubilize in urea, purify, refold, and you get pure refolded protein. In fact, while doing that, your yield is only 10%. If you are expressing 100 milligram of insulin, if you get 10 milligram pure in an industry, you are doing a wonderful job. People have been doing because the refolding is at low concentration, and in fact the recoveries are very very poor. This is to avoid this. What the people do is they do dilute the refolding. They do the refolding process at 10 to 50 microgram per ml. Remember, we express 10 gram, and when you refold in microgram, like you produce the E. coli in 1 liter, but you need 1,000 liter of buffer to refold it. In fact, if you go to a insulin plant, it is the water and urea who are the most costliest thing. They contribute majority, 50% of raw material. I'll give you one example. I do this regularly. Here I am looking at producing 15 kilos of insulin, media preparation, fermentation, all downstream. You see how much of waste you generate. 600,000 liter. And remember, these are waste with endotoxin-free water. And when you use with the recombinant organism, they are contaminated with the recombinant organism. So their waste treatment and disposal is very, very stringent. That's why it costs more. In fact, if you look at the refolding cost, really contribute to the recovery of insulin. If it is 15%, what you are recovering, it will come 30%. If you have more recovery like 30-40%, you can add to cost. In other way, the more you recover from inclusion body, the more you develop a process, you can produce the insulin cheaper way. So what is the innovation needed? This is, as I told you, this is fit for translation, but what is the innovation needed? High throughput recovery of protein. Can you improve the recovery from a standard 10% to 20%, 30%, 40%? You'll add more money because this is 30-year-old process. The technology is outdated. The patent is gone. That's why. So I'll give an example how you do. So we develop a process like this. You started a novel refolding process, isolate the inclusion body, solubilize, induce Helix formation, and promote oxidation, then cleavage and purification. You see, insulin is a 51 amino acid thing. It is a very, very old protein discovered in 1902 by William Bartik, but it is the most complex protein. And we developed this. I tell you why it is complex. It is complex because these are the A chain and B chain, and these are the intradisulfide, and this is the C chain, which forms like a seat belt in a vehicle so that it is refolded. So, and when you refold and purify, you need to cut the C chain and throw it. So the disulfide and C chain are important. And this is how the insulin hexamer thing. What we did is we went back to nature to understand how actually this is refolded. In fact, when insulin started, many people started expressing A chain, B chain, small C chain, because C you are throwing. And they succeed, but the expression level, the recovery level was not very good. So we went back and look at how the insulin is folded. In fact, this was published in Endocrinology some 7 years back. This exactly somebody has published how in vivo, in the endoplasmic reticulum, the insulin is refolded. So it comes at the whole C chain, it forms the alpha helix, the C chain is like a seat belt, then the disulfide bonds are formed, then it gets secreted. So we thought, why not copy exactly why? So our method was just like the C chain is secreted, it is refolded to a alpha helix structure. A chain, B chain come to each other. The disulfide bonds are formed. Then the C chain. At the end, you chop off the C chain. So this is how we develop. We cloned it. We express as inclusion body, and as I told you, inclusion body come as a particle. We could purify that without chromatography. This is the final inclusion body after solubilization without much of chromatography. I'm not going to detail. I'll just show you the data. And when you did that, you see this is pro-insulin, and this is our insulin showing more or less same alpha helical structure. And the real result came like that. Our expression was around 10 gram per liter. As I told you, the C chain is 50%. So ultimately, when you chop up C chain, you lose 50%. So The real insulin as inclusion body is 4 gram. We use the novel technology around 25% recovery. We get 1 gram per liter. This is what we do in 10 liter, and that means 1 liter per gram. We have 10 liter in 10 gram, 200 liter 2 gram, 500 liter will give you 5 kg. And this insulin, the E. coli is grown in 1 day, 3 days. So you can have 50 batches per year, 50 into 3 with downtime. So you can have 200 kg of pure insulin from 5,000 liter batch, and you put 5,000 liter reactor. These are very small in industry concept. From 5 kg to 1 ton, you can make it. So this is the plan. That means, as I told you, the rule, it was needed. We developed a wonderful science. We had the proof of principle because we know insulin works. We could scale it, as I told, and it is sustainable because we are not doing anything dependent on that. So this gives an idea. If you have an idea, apply these 5 things. Is it useful? Is it science is very good? It has a proof of principle established. Is it scalable? It is sustainable. You'll be able to do a translation. You may see doing what is big thing in doing in insulin. In fact, look, you can do different expression. You can still do innovation in insulin. You can do different expression system, different version of insulin, novel way of purifying, novel way of delivery. Please remember, insulin is one single molecule on which 4 times Nobel Prize has been awarded. Go back and check in the history. The person who discovered, William Bantic, the Sanger sequence was developed using insulin. The radioimmunoassay was developed using insulin, and the fast cloning. 4 Nobel Prize, one single molecule. You develop a inhalable or oral insulin, the next Nobel Prize is yours. So all I'm saying, even if it is an old molecule, it's a established process, cloning, expression, you still can do innovation and you can still do. In fact, I gave this example of doing an innovation using insulin. I work in healthcare sector, and if you ask me what are the innovations in large scale, these are my dream. E. coli has endotoxin. Can you have an E. coli which doesn't have endotoxin? E. coli doesn't produce glycoprotein. Glycoproteins are produced in mammalian cell. So the real innovation will be, can you clone the whole glycosylation system of CHO or mammalian cell in E. coli and make a glycoprotein in E. coli? Indeed, yes. Methanol-free induction, mine will be the last. You know, I'm sure again, I'm sorry, I'm going every time back to the school. In the school, you must have purified copper sulfate, like just crystallize the copper sulfate, blue crystal come. Can you purify protein just like crystallization? That is the future coming. You don't need chromatography. You know the type of crystallization. The only thing needed is you have a high concentration of protein expressed, crystallized in a condition which allow the crystal of that particular protein to be burned. Other things will be in mother liquor, just like you crystallize copper sulfate. A day will come, maybe you don't need all chromatography. Just express the protein, crystallize, and you get a pure protein. I think I'll end here. Thank you very much. You can ask me questions.
Thank you, sir, for that very simple and very, I would say, well-received presentation. The floor is open for questions now. You are having a stalwart in front of you, and you very rarely get such opportunities.
I keep coming. They will.
Thanks to Abdin, sir, that you are having this chance.
हाँ.
Will you pass a mic if possible? Thank you.
जोर से बोलो, सुनाई देगा. हम बोलो. No, no, no. See, you have to understand what is a drug or why you take something when you require. These are not food items, right? So if you are taking insulin heavy, you will have hyperinsulinemia, and all your glucose will be consumed. Then your brain stop working, and if it goes below certain level, you are gone. So never do that. First thing, and if you by mistake taken an insulin, immediately eat sweets. Either eat some sweets, रसगुल्ले खा लो दो तीन, तुरंत पिकअप करेगा, because all you have to maintain your glucose level. Certain amount of glucose level in the body is always essential to make you fit. Just remember, the carbohydrate you use is not only used for your physical things. Every time all of you eat, the carbohydrate is equally divided in three parts to do three basic functions in the body. First, to make us physically active so that you can walk and talk. Second, our brain. And third, our immune system. So you need always carbohydrate to all three things to work. I'm sure I don't know whether you know, when a pathogen attack, the first thing it does is it allow hyper secretion of insulin so that the glucose is starved and the pathogen attack because your immune system become weak. So even if you use huge amount of insulin, maintain your glucose.
Thank you, sir. Thank you for your enlightening talk.
Done?
So now I would thank you, sir, for your enlightening.
I'll go down.
So it's my great pleasure to welcome our next distinguished speaker, Professor Tripathi. Professor Tripathi is a renowned expert in the field of biotechnology and molecular plant physiology. He is a former Dean School of Life Sciences, Jawaharlal Nehru University, and former Vice Chancellor, Ravenshaw University, Cuttack, Odisha. In 2019, Professor Tripathi joined as a distinguished professor in the Department of Biotechnology, School of Engineering and Technology, Sharda University. In recognition of his scientific contributions, the Department of Science and Technology, Government of India, designated him as J.C. Bose Fellow. He is a member of the Board of Directors of the Rivis Foundation for Basic Research USA. Because of his excellent research contributions, Professor Tripathi was elected as a Fellow of Indian National Science Academy in 2005. He is also a Fellow of National Academy of Science and National Academy of Agricultural Sciences. And his research areas are in the field of molecular plant physiology, photobiology and phytobiology and biotechnology. He has also worked for National Aeronautics and Space Administration, NASA, USA. In addition to his research articles, he has contributed several chapters to books and has edited books on photosynthesis and has mentored a large number of PhD, MPhil, and MSc students. So, with these words, I will request sir to please on the dice.
Thank you very much. I'm happy to be here. Okay. Thank you very much. I am happy to be here and give a lecture. I want to thank the organizers of the conference, including Professor Abdin and other people here, to invite me and give a lecture here. And my talk today is, changing chlorophyll A by B ratio in plants. Okay. You see, I work mostly in photosynthesis, so my expertise I'm utilizing to understand photosynthesis and productivity, plant productivity. So all of you know that plants have chlorophylls, and chlorophyll, we have most higher plants are chlorophyll A and chlorophyll B, and they maintain a certain chlorophyll A by B ratio. The chlorophyll B is synthesized by a gene called CAO or chlorophyll A oxygenase. That is called CAO, which converts chlorophyll A to chlorophyll B by oxidation reaction. All of you know difference between chlorophyll A and B, where CH3 is converted to CH2. Okay. So what I'm doing is trying to increase this synthesis of chlorophyll B or decrease the synthesis of chlorophyll B in plants to see their impact on photosynthesis and plant productivity. So we are increasing chlorophyll. So to increase the chlorophyll B content, that is CAO, the gene which converts chlorophyll A to chlorophyll B, was the CAO gene was overexpressed. The chlorine become a vector and overexpressed in tobacco. And these are the tobacco plants. You can see the wild type plants. These are transgenic plants. These are at the early stage, and you can see the immature stage. You can clearly see the wild type and CAO, or the difference between wild type and the plant overexpressing CAO. Chlorophyll oxidation gene then producing more of chlorophyll B. Okay? So more is the chlorophyll B, less is the chlorophyll A by B ratio, right? Because chlorophyll B increased, and therefore chlorophyll A by B ratio will decrease. Okay? And these are the colors, these are the colors of the leaves. You can see, we grew these plants in a low light and high light, and you can see the white type plants low light, and these COX means COA overexpressing plants, which are more of chlorophyll B is here, and in the low light, and these two are in high light. Doesn't matter you don't follow low light and high light story, but if you follow chlorophyll A and B ratio, that is great for me right now. Okay. And these are we measure different concentrations of overexpressed. You see, chlorophyll A to conversion chlorophyll A to chlorophyll B is very end of chlorophyll bar synthesis part. And all these are many intermediates that lead to chlorophyll synthesis. If you see in very primitive organisms and photosynthetic bacteria, all these genes are clustered together. Chlorophyll bar synthesis genes are clustered together. Okay, one appearance. Okay, but as they were in high eukaryotic system, higher plants, they are distributed different chromosomes, they are in different parts of the DNA. But you see the regulation part, if you tinker with one gene, other genes are also equally affected. Okay. So this is a regulated network between chlorophyll bar synthesis genes. Okay, so that if you overexpress a gene, other genes also be upregulated. If you decrease a chlorophyll bar synthesis genes, other genes also be downregulated. Okay, so this is one of the interesting phenomena. So this was I'm going to tell you, but I am not going to talk about details. Here you are talking about enzymatic activity in the different plants, different chlorophyll bar synthesis genes, and you measure also many proteins, okay, many protein expression of chlorophyll bar synthesis. You can see always here, this is COX highlight or COX plus. COX plus, you get the last column. The last column here, you can always see, most of the genes were on overexpressed system, overexpressed. COX plus. Okay. Most of the proteins are high from Western blood analysis. And same thing you can see the others also, okay, other things. So most of the genes of chlorophyll bar in this pathway were upregulated in the transgenic plants. Now comes the photosynthesis. Okay, so once we increase the chlorophyll B ratio, chlorophyll B content, there are certain proteins of chlorophyll A binding protein. Chlorophyll A by B, A and B binding protein. Thylakoid membranes. Okay? That are light harvesting chlorophyll protein complex. Okay? So these light harvesting chlorophyll protein complexes, their concentration increases when chlorophyll B is more. Okay? So what do? They harvest more solar energy and transfer the energy to the reaction center. Okay? So they are able to harness more solar energy. Okay? Because they have now increased light harvesting complexes. So they can better harvest solar energy and transmit their energy to the reaction center where it is better utilized for photosynthesis. So these are electron transport rate you can see here, in the white type plants and in the in the white type plant and in the transgenic plant. Okay. You can see here photosynthetic rate here in the ratio, in the photosynthetic rate. Okay. The white type and the low light and high light don't get confused now, but we are using one simple issue, the white type plant transgenic. There's a clear cut increase in the photosynthetic rate and electron transport rate and so on. Okay. So I just go and why is it happening? As I told you, we measure did ocean blot of many proteins, and you can see LSCD1, LSCD2, LSCD3, they are all different components of light harvesting complex. And you can see most of them were overexpressed in the traffic. Okay. And similarly, you also measure other components of the photosynthesis. Oxygen complex protein 33, which is photosynthesis 2 protein, which is what oxygen. Okay. Then you have D1 protein of the photosynthesis reaction center, D2 reaction center, Nistiagan protein, cytochrome F, cytochrome A, cytochrome is nothing but C10 cytochrome, and all other different components from system 1, they are all upregulated in the presence of what's the increase of protein. Okay, and this resulted in the carbon assimilation. Okay. We did not think of this carbon assimilation. We only focused on what? Light reaction. Okay, chlorophyll A, modulating chlorophyll A and B. So electron transport increased, and the increased electron transport rate contributed to increase in photosynthesis rate, carbon assimilation rate. And these are the quantum rate of photosynthesis, both low and high light. You can see the quantum rate of photosynthesis also increased, in COX. Then we go to overexpress CaO that increase the chlorophyll B content of plant. Now, then whether the starch content during our experiment, you can see your starch content continue to increase in the transgenic plant compared to the wild type plant. Okay. And most importantly, you can see the dry root in highlight. In the white type, the black is transgenic. Okay. So you can see transgenic plants have a higher biomass than the white type. Okay. So what we therefore conclude from here in this experiment that we increase the chlorophyll B content in the plant, decrease the chlorophyll values of the plants anyway, and that led to increased light harvesting protein complex that absorb solar energy and transfer the reaction center better, and therefore they are utilized in the carbon assimilation reaction. It can eradicate and produce more from light reaction where there is no limitation of that. Therefore, Lewis-Co assimilation and carbon assimilation increase. Okay. Now the reverse part. Now we decrease the chlorophyll B content of plants by antisense expressions here. So this is again antisense plants. So their chlorophyll B content therefore what? What CO does? Converts chlorophyll to chlorophyll B. Okay. If CO is decreased, there will be less synthesis of chlorophyll B. Okay. So A by B ratio is going to increase. So we decrease the CaO content so that chlorophyll B content, chlorophyll B synthesis decrease, So chlorophyll A by B ratio increased, okay, and that led to the plants here, the wild type plants, the transgenic plants, okay. You can see the plant is little smaller than wild type. Not big, very small, but smaller. So these are different. The chlorophyll content of the plant decreased, and you can see these are all different kinds of intermediate versus pathology. But let me go, we don't have much time. So let us go to the directly electron transport. So what happens here? The transgenic plant is now here is lower than the wild type. Okay? Because wild type, the rate of capacity electron transport decreased. So because A by B ratio increased, so B content decreased, so LSC decreased, light hermetic content decreased, so less light transmission to the reaction center and less photosynthesis, and therefore you can see a decrease in the electron transport rate, and these are different light intensities, it's in the electron transport on the function of light intensity as well. Okay? So you can see a decrease, and you can see here that PS2 photosystem 2, photosystem 1, and whole chain electron transport decrease. And not only that, of course, is the light saturation curve and all that other good details, but you can see later. But you can see the expression OEC 33, D1 to be decreased, OEC 37 decreased. And look at LSC D1, this is a light ablating complete. You see the last one, antisense light. You can see now compared to wild type, all these proteins were decreased. And that resulted in reduced photosynthesis, reduced electron transport rate, and reduced carbon activity. Okay. So this is your, you can always see this is, wild type and CAO. You can see here always in the decrease in the chlorophyll rate and in the constant photosynthesis rate are different light intensity. We measure that different light intensity. 200, 400, 800, 1,000. So what it tells us? The conclusions that increase of chlorophyll being contained increases potash chlorophyll, reduces chlorophyll availability ratio, and increases photosynthesis plant life. Okay. Similarly, reduced chlorophyll being contained, decrease potash chlorophyll, increase chlorophyll availability ratio, and decrease the rate of photosynthesis. So modulation of CO2 expression is going to increase plant productivity, and then the group of China will come out to a procedure in the right, and so that if you overexpress CAO in the right, you can get better biomass and better energy. This is one thing that I must tell you, and thank Dr. Potnak for the research in the country for contribution. Thank you very much for coming.
Thank you, sir. Thank you for your enlightening talk on the expression of glorified A oxygenase. So the session is open for questions, please. Any questions from the audience? Thank you, sir. We are honored to have you here.
Thank you so much, speakers and chairpersons and participants. Now may I request our chairperson to present the mementos as a token of respect and appreciation.
So I'd like to conclude the session, starting from our most favorite speaker, Professor Omar Pandit. We would like our family member, always being in the department to support us from our board of studies and all. I remember our syllabus and all. You have, given all your efforts, fermentation technology, everything, all your inputs. You have contributed a lot to our department, and whenever we have any need, we always rely on you. Thank you so much for your enlightening talk today on translating innovation in biotechnology. Your explanation, I must say, always it's very simple and clear. so that all students can get the benefit of that. Thank you, sir. So, and I will say I'm also for our other distinguished speaker. So we are really honored to have you so senior professors in our gatherings and our students. I am confident and I'm very sure that all of them have benefited from your talks. And we like to have more conferences in future and hopefully have both of you here with us again. Thank you so much.
I think you have captured well all the parts and all the aspects of the deliberations. And I'm extremely happy that the presentations, both of them were of class A, nothing less than that. One was very research intensive, so it gave us a lot of hope at what could be the research areas and ultimately aiming to have a productive farming or a productive crop. The first speaker primarily again spoke very well simplistically as Dr. Humaira pointed out earlier. And as a student, I am taking home a lot of lessons, which I am sure that it is a shared idea. So what is needed, what is reproducible, what is validated, what is scalable, and what is sustainable. This whole conference is in and around sustainable, and I think the department is already aligned to the SDG goals. Thanks to the leader at the house and also the organizing committee for bringing this topic up. And needless to mention that the example of insulin was very at-home example, which everybody could relate, and how the need has to be actually commensurate with the production, and how you people as young biotechnologists can actually fill that gap or meet that unmet need. So the slogan which we have been hearing since ages, like in our childhood, was like Jai Jawan, Jai Kisan. But the new slogan is Jai Jawan, Jai Kisan, Jai Vigyan, and Jai Anusandhan. So we cannot do anything without the Anusandhan and the innovation, and that was very beautifully deliberated. Thank you so much for this opportunity, Abhi sir, and the organization.
Now may I request our chairperson to present the moment to our speakers as a token of respect and appreciation. Dr. Amulya Kepada sir.
This place has
And to present the memento to visit the party, sir. Sir, जी. Ma'am, please. Now may I request our organizing secretary, Professor M.Z. Ravni sir, to present a mementos, our chairperson, as a token of respect and appreciation.
Now I invite all the speaker, chairperson, moderator, and organizing secretary, sir, for a group photograph. So for a group photograph, please. Now it's the time for tea break. I would like to request all our guests to join us for tea. We will resume after 15 minutes with technical session 4A. Thank you.
He contributes to food safety, polymer science, and industrial radiation applications. The topic of his presentation is role of radiation processing for food safety and security. May we have a round of applause for Dr. Sanjay? Over to you. So the presentation is for 20 minutes.
So, good afternoon. First of all, I would like to thank Jamia Ahmedabad University for inviting me in this very important event. So, I would be basically talking on your application part, the applied research, especially the role of radiation in various industry sector. And here my focus would be the for food safety and the food security. This food safety and security of the nation determines its economic stability and the self-reliance, the fast-growing population and the shrinking arable land, increasing costs, agriculture inputs. We should not only aim to increase the productivity, but also to look into the technology to preserve it. The major concern in India is, as per this FSSAI and the report and UN report, in 2020, the Global Food Security Index, India ranked 68th out of 113 major country, and in 2023, Global Hunger Index 105th out of 127 countries. The impact of so many regions are there. One of them is the impact of climate change, and as a result, 43% of the children are chronically undernourished. And this is basically the low awareness of the food safety needs. And as per this FSCCI report, there is an increase in the adulteration of the food basically. In 2002, it was 15%, now in 19 it was 28%. It is because of the poor traceability, supply controls, hazard management, and lack of affordable healthy food, lower agriculture productivity, lack of infrastructure, and as a result, 100 million foodborne diseases every year. And because just to control it, the Food Safety Standard Authority has been established in 2006. For food safety and security, there are parameter and there is scale basically. For production, you need to improve the productivity, and then second is the availability. For to make it available, you need to have a preservation techniques, and for sustainability, you need to maintain the requirements, and for quality aspect, you need to have a safety aspect. And maneuvering these factors, the above factors, their of desired scale can be achieved by the irradiation technology. What is the food irradiation process actually? It's a very simple process. It is the food is exposed at a specified dose, at a particular dose rate inside the biological chamber, biological shielded chamber, and food irradiation. These food goods are conveyed to the irradiation chamber inside the room through, and it is controlled from outside through SCADA system control panel, and conveyor system is used through variable frequency drive at a particular speed. The speed can be adjusted as per the required radiation dose, and it depends on the source strength also. And cobalt 60 basically the nuclear material is used for this particular purpose, and dosimetry is carried out to measure the absorbed dose by the material. There are different type of gamma ray. The gamma ray is basically the 1.33 million electron volt, and 1.17 is the energy level of this. And these are the recommended source basically by the WHO, FAO, and IAEA. So this is the just overview. This material goes through conveyor belt inside the chamber, and this is a biological shielding around the source, and it goes for a fixed period of time for a desired radiation dose, and it automatically it will come out through conveyor system. This basically food addition process is a non-additive process as compared to the other techniques like ethylene oxide or other treatments, and causes minimal changes in the food, preserve food in the natural form, and is a cold process. There is no heat involved in that technique. And the most important characteristic of this cobalt 60 is a very high penetrating actually. It can penetrate throughout the material, or the big size of, you can say like 2.5 feet thick, big size of box can be processed at one time, and can be applied the final product. That is the beauty, another beauty of the product. Just make a product, sealed it, and in the final pack it can be processed, and whatever the mycological contamination inside the material, it will be taken care, and does not leave any harmful residue since it is no chemical is used, and safe to the workers. This is basically a purpose based application and a single process for quarantine treatment. I will just explain you where it is important for any country, for pathogen reduction in various spices and other items, and shelf life extension of these various climatic fruits like apple, and then insect disinfestation in spices, sprout inhibition, onion, potato. And it is categorized on 3 category basis: low dose application, medium dose application, and the high dose application. For low dose application, disinfection, basically disinfestation of the insect, inhibition of sprouting for tuber bugs, delay in ripening process, and the medium dose, the destruction of the microbes responsible for the food spoilage, meat and meat product, poultry, marine product, hygienization of spices, and then high dose, elimination of the parasite pathogen for public health. This technology is basically plays an important role in post-harvest management actually. Because of the poor post-harvest management, because of this inadequate harvesting, pre-packing, handling skills, lack of suitable packaging, inadequate storage and the transportation facility, and lack of technical management capability, the significant loss in agriculture produce is due to the insect infestation, mold, premature germination, and sprouting. And gamma radiation, cobalt 60 can play important role in reducing post-harvest losses. And in our country, so many plants are there. They are providing these services to the food processing industry and the agriculture producers. I would like to just give you a brief idea about the status world over about the food processing in various countries. And this China is basically has a maximum number of radiation plant where they are doing a lot of food radiation. More than 500,000 metric ton annually they are doing the food radiation for various products. Like in USA, this approximately 80,000 metric ton of spices are being processed through gamma radiation technique for various purpose-based application. And this is just, I would like to give the idea of this status of the various countries where these food products have been approved for various purposes. So this is the list of the country. This is very important application of this cobalt 60 basically for quarantine treatment. Like any agriculture produce of fruits, vegetable, if you want to export your material to any country, so there is a USDA framework where it has to be processed through gamma radiation. Otherwise, you won't be able to export the material. And in USA, 21,000 metric ton annually they exporting. And this is the member country of this framework actually. Only this country can exchange the import export because of this. They have the radiation processing facility, and India is one of them. We have a good capability about this nuclear material, handling of the nuclear material and the radiation processing of the food product and other item also, other industrial sector also. So this is the status of this permitted fruit and vegetables, especially the climatic fruit for phytosanitary treatment. So this is one of the main requirement for any especially this US and Europe and other countries, they are importing this material only after radiation processing. This is the one success story of our country. This mango, export of the mango was banned actually by US because of the seed weevil infestation. And this food technology division BRC, they had developed a technology of this the seed weevil disinfestation through gamma radiation technique at very low dose. And because of this, now we got the approval, and we are able to export mango after radiation processing to US and other countries. And in 2023, we had exposed around 27,000 metric ton of mango. In India, basically, we are processing mango, pomegranate, onion, potato, spices, seasoning, dried vegetables, herbs, and dry fruits. Various plants in different places in the country are there, and 50,000 metric ton annually is being processed. It's still very low quantity. And this is just giving you an idea that there is radiation plant at various locations in our country. This using the cobalt 60, and 18 more multi-purpose plants are coming for radiation installation of medical devices as well as the food processing for various purposes. Then these food product, basically radiation processing of food product are covered by FSS, Food Safety Standard Authority of India, and they have given a guidelines basically, and they have classified the these fruits and vegetable and other food items as per the category for various purposes. They have defined the radiation dose also, dose limit, the minimum dose and the maximum dose for that desired purpose or benefit, technological benefit actually. So we have some experience in radiation processing of food. Like I worked in this self extension of the apple using cobalt 60, and then bamboo shoot is one. Then disinfestation spices, wheat flour, pulses, gram, dehydrated vegetable, herbal product, dry fruits, meat and meat product, and removal of aflatoxin in peanuts. So I will just like to share one or two slides about this outcome of this self enhancement of apple using radiation technique. What we did actually, we process apple at the particular maturity level, because what happens, this apple this ripening of the apple takes place during storage process, and it is temperature dependent. Like after maturity it is plucked, and then it is stored in the cold storage at 90%, 95% humidity and the 2 to 4 degree temperature, and it can last long for 4-5 months. After that it start decaying. And if it is processed through gamma radiation, then you put in the radiation, this cold storage, it can be, this sulfur can be enhanced by 3 or 4 months. So what we did, we processed at different radiation doses, and it was kept at two temperature conditions, and the various parameters were evaluated: physicochemical, sensory evaluation for taste, texture, aroma using 9-point hedonic scale, then microbiological evaluation on the surface. And what happened actually, at the time of maturity, apple has basically carbohydrate, which is converted into sugar during the ripening process. So after radiation processing, what happens is it delays the ripening process. So as a result, sugar conversion would be delayed. So this is just I'm just comparing the control. The sugar conversion in the irradiated sample was less, also maintained in irradiated sample as compared to the control sample. So just see this. This is the golden variety of Himachal Pradesh, and you just see the even the color is retained after radiation processing, even after 8 months of storage at 4 degrees centigrade. And control sample is just color is changed, completely matured. Like we also studied this the self life at the ambient condition of the Himachal of the 15 degrees centigrade. And unirradiated apple start after 3 months of storage 15 degree, whereas this irradiated sample, it retained 5 to 6 months. The outcome study was this: the irradiated sample and its properties were maintained even after 8 months of storage, whereas unirradiated control sample start decaying after 3 months, 3 to 4 months at 4 degrees centigrade. So there is a how much? 2 minutes? 5 minutes. So uh, 3 to 4 months self extension was achieved in this project study. Then I'm just going to share some slides about the effect of gamma radiation on various food item. How is effective to reduce the microbiological load in the product, in the material. Like we use this radiation processing for decontamination of the dehydrated carrot. So even it is significantly completely removal at 3 kilogram dose, which is a low medium radiation dose. Then we also check the nutritional parameter which were intact. There is no significant change in the nutritional parameter. Then we also did some study on removal of aflatoxin using radiation technique, and up 8 kilogram dose it is totally removed. And we also carried out these chemical properties of the peanut, and there was no significant change up to 8 kilogram dose. Then we did decontamination in the herbal product. Various types of herbal product we just studied, and it was found that very highly contaminated herbal products or Ayurvedic products, and they at 5 kilogram dose, which is a recommended dose, or which is basically the dose approved by the FSSAI or other countries. And at 5 kilogram dose, the complete removal of the biological was found. We did the total viable count basically. And we also did one study on lychee, and again it was a very low dose application at 1.5 kilogram. There is a significant increase of around 2 to 3 weeks. The irradiated lychee were found in good condition even after 24 days of at 4 degrees centigrade, whereas the other, and also did the sensory evaluation, and we have published this study in the radiation physics and chemistry. And we also did on bamboo shoot. There is a limitation of food irradiation basically. Not, it is not suitable for all types of food. Like if food is spoiled, it is not recommended to process it. Then the the product which have a fat or dairy product, foods with high water content is also not suitable for decontamination purpose. But where is the climatic fruit, the purpose is different, just to check the ripening process. So there it is useful, and not possible to eliminate the pesticide or other chemicals. And because these cost is also increased, but at the same time synergistic effect can be achieved by using the other technique along with the gamma radiation. So in that case you need a comparatively low dose, and you can some damage in the product can be avoided. There is again a issue of the consumer acceptability of the irradiated food, and due to the lack of awareness about the safety aspect of irradiated food, it is less preferred than conventional processed food. And once the truthful importance about the irradiated food and its purpose is established, consumer in various countries will prefer irradiated food. And encouraging result of the market trials at various places, different countries led to successful introduction of the irradiated food at commercial scale, like Belgium, China, France, Japan, South Africa, Thailand, and India. And also we are in India also, BRC Baba Research Center has done. This is a safety aspect of the irradiated food is a big issue. Like, so in the last 5 decades, a lot of data has been generated on radiological safety issue, the safety of chemical changes, microbiological safety, toxicology, and nutritional adequacy. And it was approved that up to 10 kilogram it can be processed without any use. So a lot of data is already generated for that, and it is safe up to 10 kilogram. And that's why there's more than 60 countries have approved the irradiation for various food. Then I would just give you one idea. It also plays an important role in biotechnological research, produce high yielding, high protein containing varieties disease-resistant varieties through mutation, and Bioatomic Research Center, they have done a lot of work in this area also. And we also did on chili powder, is a significant. Sorry, actually, uh, last slide, Mr. Kadam, last slide. Okay, sorry. The way forward, actually, the irradiation provides an added layer of safety to many food products which are susceptible to contamination by pathogenic microorganisms. And recent trend indicates that irradiation can play an important role for food safety and the food security of agro product. And due to strict content regulation, food irradiation is bound to be useful means to take care of the trade barriers in export of agro product. And more awareness program of the safety benefit of the irradiated food product need to be created among the consumers, and more research need to be conducted to enlarge the list of the irradiated product. So we can include more food item in that list. There is a requirement to explore the possibility to use the irradiation technology along with the other processing technology to get the synergistic effect. And this one more important information: this any food product, when it is processed through the gamma radiation, so one has to label that it should be a regular. This regular symbol is there, so it has to be pasted that it shows that the product is irradiated through gamma radiation or electron beam. Okay, thank you very much. Yeah.
We can take a couple of 3 questions. Before, I have got a question for my understanding and understanding from the audience that how impacts the irradiating the fruits and vegetables? Does it impact the quality, like what we eat?
Yeah, that actually depends on the application part. Like it is not recommended to do on the fresh vegetable. Okay, there's one aspect. It is used on the dehydrated vegetables, which where the moisture content is not more than 10%, less than 10%. In the product vegetable which have more this moisture content, the radiolysis can take place. So it is not recommended in that case. But at the same time, where the ripening like climatic fruit vegetable like apple is a climatic fruit, so their ripening takes place during the storage, so enzymatic reaction can be retarded through radiation at very low dose without affecting the sensory and the other important properties. So it depends on the application part, but not recommended the fresh vegetable. Yeah.
Any other question from the audience? Yes, please.
Afternoon, sir. I really enjoyed.
Sir, I would like to ask, are there any pen-based probes to self-check the radiation limit in fruits or vegetables you buy in the market?
I just mentioned that. Can you repeat, please?
Sir, are there any pen-based probes or that we could find, like if we need to measure the quality of a diamond, we use a pen-like probe to check the quality. So is there any limitations to as the radiation limit and that probe
Yeah.
which we can use for us?
Like I just explained you this the radiation processing that is done, So certification is produced at the time, means ready how much radiation dose is absorbed by the material. Like if it is a spices, so 6 to 10, approximately 10 kilogram average dose is approved. So it should not exceed that 10 kilogram dose. So this speed of the conveyor is adjusted for that dose only. After that, there is a dosimeter, steric Saras dosimeter. That dosimeter are placed along with the product, and the certification is provided along with the processed material. So that certifies. Secondly, if you have a doubt, another thing is the whether product is irradiated or unirradiated, you want to check it. So there are techniques for that to check this. Yes, because the free radical are entrapped in the dried matrix, and it can be evaluated through techniques available. So one can check this product is irradiated or not irradiated.
And sir, I would like to ask, are there any hypotheses or prospectus like if we take daily some amount of radiation food, will it alter something in the future? Our genome or any other?
No.
Small radiations every day will amount to a large quantity.
Okay. No, that's why these guidelines are there basically based on the product and the radiation dose. So I just showed one slide up to 10 kilogram dose, which is very high for fruit. Like just to check the sprouting, only 0.05, 0.1 kilogram, very low dose is given. So at that dose, there is no change in the properties, and 10 kilogram dose is safe from radiological, toxicological, all point of view. So that's why this was approved by various countries.
Thank you, sir. Thanks a lot.
Thank you very much, and I'm really thanks for the speakers that they have set the very exciting tone of the session. And before the going to the next speech, the next our presentation, I would like to call upon our chairman of the session that they should please to please felicitate Professor Shailja Singh for nice presentation and setting a very good tone of the session for this very knowledgeable session, and for very exciting in a very exciting tone she has presented. So thank you very much. Thank you very much, Dr. Shyam, very much, Dr. Shaukat Saab, and thank you, Professor Shailja Singh. Before moving to the next presentation, I would like to call upon Dr. Shaukat Hussain to introduce the next speaker. Our next speaker is Dr. Shyam Ali.
It's a really chair of the session, Dr. Shyam Prasad. She is a professor in the Department of Medicine at New Delhi. His research focuses on molecular biology, immunology, biochemistry, with specific interest in PCOC, hepatocellular carcinoma, bone carcinoma, and viral kinetics. He is a fellow of IABS and has been awarded with Presidential Award on Digestive Disease. Professor Nunu Award Dr. Prakash has published extensively in high impact journals. The topic of his presentation is a perspective of transfer RNA-derived RNA fragments and mitochondrial functioning in metabolic dysfunction associated steatohepatitis liver disease. Over to you.
Thank you. Thank you for the nice introductions, and thanks to the organizers and Dr. Humaira for the giving me an opportunity to speak some of my work. Now I am going to speak on the perspective of the transfer RNA-derived RNA fragments and mitochondrial functioning in the metabolic dysfunction associated fatty liver disease. Now today is the fatty liver disease is a great concern globally as well as in our country. The problem is here we don't have any medicines for the treat the patient of the non-alcoholic fatty liver disease. How that the non-alcoholic fatty liver disease is progresses. It starts from the simple steatosis, then it goes for the steatosis, then it goes for the steatohepatitis, then goes for the cirrhosis, and then for the hepatocellular carcinoma. There is a progression stage. Now, as per my previous work, we have seen that most of the patients, they are coming from the diabetes as well as in the cardiometabolic disease. So the why the people's why the patients are coming for the diabetes as well as goes for the cardiometabolic disease. So in this way that we have seen that, then based on that the terminology has changed, metabolic dysfunction associated fatty liver disease, because that is the metabolic disease. So the global scenario, if you look at the NAFLD, is the 25.24% and 40.76% progression is leads to the fibrosis. If you look that it is a multifactorial issues, such as the dysbiosis of the gut microbiota, insulin resistance, hepatocyte injury, inflammation of the adipose tissue, environmental factors, ballooning cell swelling, obesity, genetic factors, reactive oxygen species, dyslipidemia. So the steatohepatitis is a liver disease, a clinical pathological syndrome, we can say, that marked by the excessive accumulation of fat, means more than 5% of the fat deposit on the hepatocytes, which leads to the hepatocellular carcinoma and rapidly growing third leading cancer related to death, 800,000 deaths annually. If we look at the natural history of the non-alcoholic fatty liver disease and metabolic dysfunction associated fatty liver disease, the NAFLD patients, from the 20 to 30% patients, they 3 to 5% are prone to develop the NAS, and then out of them 20% they are the cirrhosis, and 5 to 10 years leads to the liver death or end stage of the liver disease. So similarly, if you look at the how that the progress has fast in case of the metabolic dysfunction associated fatty liver disease, 38.8% MAFLD, around they are the metabolic steatohepatitis, that is the 25%, and 49% cirrhosis and liver death. Here is the problem is here, because once the patient is establishing that the metabolic associated fatty liver disease to steatohepatitis, the cirrhosis we are missing that, and that patient directly goes to the end stage of the liver disease. So if you look the grades, various grades of the steatosis, simple steatosis, mild steatosis, moderate steatosis, and severe steatosis. And then stages we looked at F0, F1, F2, F3, and F4 category. So the problem is here, because liver biopsy is a even we know that liver biopsy is the standard protocol, but it's very difficult to ask the patient. Every patient, they know that they are carrying the grade 1 and grade 2 fatty liver disease, but we are asking if to come for the liver biopsy, they are not coming. There is a, we are also be missing it here. So that so we are thinking that how to develop that the non-invasive scores. So there are the various non-invasive scores, so such as the NFS, APRI, FAST score, BARD score, and FIB4, that can serve the purpose qualitatively for the diagnosis of the liver fibrosis from grade F0 to F4. That here, that only thing, the problem is here, the sensitivity of these score is not more than 70%. So fibroscan is one of the option. There is a semi-quantitative scanner that can help to assess the grading of the liver stiffness, fat deposition status, and relationship between the steatosis and liver fibrosis. But here we are also be missing for their sensitivity and accuracy. So however, many other risk factors such as weight, waste, hyperlipidemia, fasting blood sugar, uric acid, AST, ALT, CBC, they are involved in the progression of the steatotic liver fibrosis. So we can say it is a lifestyle associated disease. So societal lifestyle change, diet and exercise is of course there is a one of the important concern. Then increased fat mass, that is this expansion of the adipocytes, then insulin resistance, type 2 diabetes, central obesity, hyperlipidemia, and hypertriglyceridemia, and NAFLD or MAFLD. So if we look in this picture it here, this is if we are doing the taking the healthy diet and physical activity, that the adipose tissues are the very closer to each other. But if we are taking the excess food and we are not doing the physical activity, so here the expansion of the adipose tissues are here. So if that adipose tissues are the increasing, so increase the pro-inflammatory cytokines, increase the triglycerides level, and so we can say the chronic low grade inflammation. So based on that, we have thought about that there are the due to the multifactorial issues, so immune cells and inflammation interaction, how it occurs. So there are the various free radicals you have to look at here. There is the adipose expansion. Then here is the some genes we have looked at. There is a immune cell CD4 and CD8 cell, and then how that the fibrosis is involved in the progression of the NASH. So another point is here we have also checked that the metabolic reprogramming from the progression from the steatohepatitis to the hepatocellular carcinoma. If you look at here, there are the various genes which are involved in the progression of the hepatocellular carcinoma. Even if you look at here, the oxidized LDL, that oxidized LDL is the they are the collagen deposition, and then inflammation is increasing. Then similarly iNOS dysregulation, ADMA, and ADMA, and then it goes for the hepatocellular carcinoma. Then another aspect we have looked at the how that molecular interactions which occur. High fat diet, high fat diet, sugar and free fatty acid, insulin and insulin resistance, and then how they are infecting to the liver, and that then how that the mitochondrial dysfunction as well as the involvement of the transfer RNA derived RNA fragments. Idea is that we are searching that how we can find a biomarker that can tell us some information the to we can stop at the early stage for the progression of the hepatocellular carcinoma. So and then how that the mitochondrial dysfunctions are involved. There are the various proteins, mitofusin and mitopherin proteins, they are involved, and how they are triggering the molecules of the transfer RNA derived RNA fragments. And there are the various formulas to we have to be calculated, but I have already told to you that their sensitivity and accuracy is not more than 70%. So we have targeted some genes. GIP8 is a multifunctional membrane transporter that influx the essential metal cations and non-metal inorganic selenide. GIP8 is involved in the regulating of the redox balance. And then hepatic GIP8 dysregulation are the major concern in the liver pathology, that is which is involved in the oxidative stress, inflammation, proliferation, and hepatocellular injury. And even you can say that the multiple biological mediators which are involved. So another molecule we have targeted the prokineticin. Prokineticin is a small peptide that have many activities in and involved in the proliferation, migration, differentiation, and which is involved in the angiogenesis and muscle contraction and immune response. So another portion I took that the mitochondrial fusion is mediated by the mitofusin protein 1 and dynamic related to the GTPase that is responsible for the fusion of the outer mitochondrial membrane. Increased mitochondrial fission and the decreased mitochondrial fusion indicate mitochondrial fragmentation. And another, we have looked at the eNOS are involved in the many pathological state in patients and has recently been linked to the occurrence of the metabolic syndrome. Another part we have checked out the GATA4. GATA4 is a zinc finger transcription factor in the liver sinusoidal endothelial cells, and it plays a critical role in the liver regeneration. And then another molecules we have checked out the how that the platelet derived growth factor B and their role in the pro-fibrogenic trans differentiation of the hepatic stellate cells during the hepatic fibrosis. So metabolic programming, I have told that there is some dysregulation of in the glucose, lipids, and amino acid metabolism. As we know that the Warburg effect marked by the increased glycolysis facilities rapid energy production and biosynthesis of the cellular components into the hepatic cells. So amino acid metabolism, particularly glutamine, utilizes the supplies critical carbon and nitrogen for the nucleotide synthesis and maintain the redox homeostasis. So transfer RNA are generated by a ribonuclease, including dicer and angiogenin, participants in many biological process. miRNA and transfer RNA derived RNA fragments, small fragments, are the essential transcriptomic signatures that can be applied as a non-invasive therapeutic tool for an early assessment of the liver steatotic severity in the NAFLD or MAFLD patients. So abnormal expression of the transfer RNA derived RNA fragments is found during the prematuration and maturation state and has recently been reported in few cancers such as the colorectal cancer, prostate cancer, even in the breast cancer. So if you look the structure of the transfer RNA derived RNA fragment, non-coding transfer RNA, which is derived from the precursor or precursor or the mature transfer RNA. As we know that where is the RNase Z from the 3 side and RNase P is work and their fragmentation occurs of the transfer RNA derived RNA fragments. Then if you look at the relationship of the transfer RNA derived RNA factors and spectrum of disease, how that the transfer RNA upregulation and downregulation is involved in the various metabolic diseases. So the question we can say is the insulin resistance is the major one, alteration of the glucose and lipid peroxidation level, that alteration in the arginine, glutamine, enzyme and formation of the new antigen and dysregulation of the GIP8 or GIP14, it increases the oxidative stress, increases the advanced glycation end products and increases the cytokine and eNOS and iNOS dysregulation and prokineticin. So that is the increasing the fatty liver and diabetes are the metabolic aberration. So these are the data we have done. We have took it here the pure metabolic disease, we have categorized the diabetes, metabolic syndrome, and diabetes with metabolic syndrome, NAFLD and MAFLD patients as per the various guidelines. Then oxidative stress and antioxidant stress, we have done that. Lipid peroxidation, we found that the very higher and superoxide dismutase was the down at the down regulation, we can say that, and ferric reducing antioxidant potential was also is the from the healthy control, it was decreased. So adiponectin, as I told that the from the muscle it is released that adiponectin, adorpin, prolidase, IL-1 beta, IL-6, TNF alpha various cytokines and adipokines we have done, and we found that how they are involved in the progression of the disease. And if the BMI is increasing the relative index of the diabetes risk and obesity. So atropine levels, I have seen it here in the NAFLD from the healthy control. If you look the NAFLD from the healthy control, it is decreased, and adiponectin is also from the decreased level. And prolidase in the NAFLD and MAFLD, it was increased as compared to the healthy controls. Then strength of the association, I have checked that. We noticed that the positive correlation between the different level of the different cytokines and insulin levels. Then different grades of the cytokines we have compared with the different grades of the fibrosis as well as different grades of the liver disease, and then we found that how that the association of the cytokines are increasing with the severity of the disease. Then the gene expression we have done in the various cases such as the MAFLD, NAFLD, diabetes mellitus and healthy control. Then we have found that the prokineticin level was the as compared to control. Yes, I'll finish very fast, no problem. Then I have seen the eNOS and iNOS mRNA expression. iNOS expression and eNOS expression, I have seen it here in the NAFLD and MAFLD. It was the upregulated GIP8 mRNA was the as compared to the NAFLD and MAFLD higher. Then NRF2 gene was the decrease in case of the NAFLD and MAFLD. So mitochondrial proteins and cytokines we have done it here, and we found that the how that the MFN1 and MFN2 and IL-10 was involved. Then T cells of subsets of we have seen that the Treg numbers are the decrease as comparison to the healthy controls. Even then here we have done that the immune cell markers with the cytokines, and it's clearly shown in the correlation how these the Treg cells are involved. The blue color is telling that how the correlation it has the severity of the disease. Then GATA4, here we got a very good relation with the GATA4. GATA4 is the increase and PDGF was the decrease. There is a one hypothesis we got it here. If the liver is going for the end stage disease, then the GATA4 is decreases and the PDGF beta was increased. So the different transfer RNA fragments we have done that. Then we found that how that the transfer RNA fragments are involved in the various category of the diseases. Then we are concluding here, decrease the insulin resistance, then decrease the formation of the aldehyde and advanced glycation end product, and the increase the nitric oxide and improvement in the endothelial dysfunction. Then we can look, we can say that yes, how that the MAFLD or NAS, they are involved by the various dysregulation of the gene. So calorie-rich drink and inadequate exercise are alarming state which are leading to the metabolic defects. Regular exercise and the nutritional bimetal supplements that could potentially limit the inflammation and reduce the risk of the chronic metabolic disease. GATA4 expression was significantly lowered, while PDGF was significantly higher in the NAFLD associated SCC. Increased arginine in cirrhosis to the SCC, that is one of the also be essential to make an understanding how to slow down the development of the disease. Then prokinetin and atropine levels could be a potential tool for predicting of the development of the NAS. Urgent need to develop the methods for the transfer RNA derived RNA fragments in the NAFL patient to predict fibrogenesis risk and development of the therapeutic tools. So by translating above insights into the clinical practice and public health initiatives, the exploration of the transcriptomic target-based nutrition can be planned for the metabolic disease. So small RNA sequence, we have found that TRF, valine, CSC, and TRF, alanine, CGC, and TIRNA in his tag were upregulated in the as comparison to the liver biopsy. Similar we got from the plasma and liver biopsy. So metabolic reprogramming in the liver cancer, gradually uncovering the ways to support the survival and proliferation that could be a scientific foundation for the metabolic therapeutic strategies. Thus, need for the combining approach into the metabolic regulatory network, liver cancer will support more accurate disease classification, innovative metabolic therapies, and improved outcome. So in this figure, you can look at increasing awareness among the healthcare providers, media and social network to the increase general public awareness, early detection, especially at the high risk group, and then it has to be make the policy to encourage the dietary changes. And these are our teams, gastroenterologists as well as the medicine, and that other my scientists and PhD students, they have worked all this to make the work for the success for that. Thank you so much.
Thank you, sir, for the wonderful presentation. So, we're just wondering, not see the role of AFP in AFP along with.
These we have already done.
Yeah.
That be our screening.
And what about the role of APC?
Yeah,
may.
we have screened carefully because we have made that hepatitis B and C with a separate group. Those who have developed the method. So we have made that different categories. There is a lot of the effort to explain.
Are there any potential questions before we conclude the session? Right. No? Yeah, please go ahead. You are very active. No, ask.
Then the GATA4 which you mentioned, which help in liver regeneration, are there any other potential aspects where it can also be used?
See, I was trying to look in that there are various GATA.
Kata 1, kata 2, and kata 4, and kata 5, and kata 6. So we have scale over, then we found that there is the kata 4 and for the carbogenesis development. So in this way, I took that, and then another we took that platelet derived growth factor B, because the platelet aggregation that will be one of the important to develop the fibroblasts. Sir, the other data which you mentioned, the fourth one is more beneficial. Yeah, that's why we found that there is a one pathway to go to understand the pathway how that the liver cirrhosis is progressing. From the simple steatosis, then steatohepatitis, then goes for the cirrhosis, then goes for the hepatocellular. So that different category we have made for us. You mentioned there, healthy those, but they like screen that they have a within the limits. So how do you define the healthy control? See, that's right. In this study, we took that the pure healthy control. I have screened around 200 medical students, MBBS students. So out of them, I found only 30 students. They are coming under the in the healthy control. That was a big challenge for us, and we have put a lot of effort together. So with this, I thank all the speakers and my co-chair. So over to you.
Thank you very much for a very exciting session. And I'm really very thankful for the Dr. Shyam Prakash for chairing as well as presenting, and Dr. Shaukat Hussain for chairing the session. And with the smooth conduction of the session was very nicely arranged by the chair. And I also thankful for the presenters who presented very nicely. Dr. Sanjay is here, and Dr. Shalja, I think she left. So with this, I just would like to request our chairs that if they want to conclude the session with some 2-3 bullet points for the as the conclusion of the session.
Suggest is like to conclude Dr. Shyam's because it is close to my heart. That healthy diet and regular exercise can keep the metabolic disorders at bay. So that is one of the key messages. And also, after listening to Dr. Shelja's talk, that there are advances because in ICMR we have a centers across the country, that National Institute of Malaria Research. So it was very wonderful to know, heartening to know, the advances in research. So it is a good piece for the students, especially who want to undertake the research in malaria, metabolic disorders, and also very importantly by our one of the speakers who mentioned that how food safety and processing is important. So so together with that food, so it comes to being a cancer researcher, I would say. So it can prevent not only the cancer but also other diseases. Thank you.
Thank you very much. Thank you, sir. Dr. Prakash, would you like to say something for the conclusion of the session?
Yeah, I think the lecture was all the lecture exciting and informative. So that is good for this conference. I have seen it here, even though I have joined very late, but I have seen that the two lectures, and there are some more informative for that to understand that. Even here told the one Dr. Rawat,
Dr. Sanjay.
Sanjay. Dr. Sanjay has told that there is a irradiation, but the irradiation, there are the how that the if you are giving the radiation, then is it affect the micronutrient? Because that is the one of the
Actually, it's in the last 6 decades, a lot of data has been generated on the various aspects of the nutritional, toxicological, and other. So, keeping that in view, there are various regulatory agencies in our country, FSSI, they have mentioned the safe radiation dose at up to that level, if radiation is provided, all the nutritional parameters are intact. That's why radiation is not cannot be given to all products like food, milk product, fat. You can't give that. It will affect. Then of course there is vitamin C is one. Then if you go to the nutrition value, okay. If we process at a high dose, if more than 2 kg or 3 kg of 5 kg, it affect this vitamin C. Vitamin C will be reduced by 10%, 20%, 30%. But at the same time, the other techniques are available for food processing. There are also a lot of nutritional value are lost. So it is based on the data generated. The nutrition dose have been prescribed, and that's why purpose is like that 0.2 kilogram, 0.3 kilogram for onion and potato. No nutrition loss.
So is it safe for the irradiation?
Yeah. Then we come to the toxicological aspect. So the safety aspect, a lot of data has also been generated in the last 6 decades. That's why these upper limit have been defined for each product. The 10 kg WHO and FAO, they have approved the 10 kg dose for radiation processing of various food products. And it is basically purpose.
Yeah, thank you very much for this wonderful and informative session. And last, yes.
For you, so I would like to personally thank Humera ma'am and Saima, Dr. Sohail. Actually, who was the person who actually introduced me to Dr. Panda, and it was very well organized. And congratulations to you all. So, can we have a round of applause to all the organizers for thank you. Thank you.
You did my work very easy. Thank you very much, and thank you for very informative sessions. And before I just conclude the session, I would like to invite our dynamic leader, Professor MZ Abdin, and our very polite HOD, Dr. Humera ma'am, to please come on the dais to facilitate our presenters as well as our chairs. So, for the felicitation of Dr. Sanjay Rajput, please come on the dais. And I would also request to the Dr. Shyam Prakash and Dr. Shaukat Hussain to please join to felicitate our speaker. Dr. Sanjay Rajput, thank you very much. Please, Sanjay Rajput, Dr. Sanjay Rajput. Dr. Sanjay, it's for you. It's for you.
It's for me.
Yes, sir, it's for you. Dr. Sanjay Rajput. Now I request Dr. Professor Abdi Saab, Dr. Humera ma'am to please felicitate our chair and co-chair Dr. Prakash and Dr. Hussain. First is Dr. Okay, Shyam Prakash. So for Dr. Prakash. Please felicitate our co-chair Dr. Shaukat Hussain. Thank you very much for everyone and you all people who are very patiently listening the session. Thank you very much all.
I would like to request chairperson to felicitate Dr. Shehzad Ahmed with the memento. Now I request all the speakers, chairpersons and Dr. Shehzad Ahmed for the group photo.
Please, sir. Dr. Shaukat, please. Thank you very much. Thank you.
So here we are done with the scientific sessions. The next scientific session will resume tomorrow at sharp 9:30 am in the convention hall number 1. And we have organized the culture program for you at 6 p.m. today in this hall. Thank you for your attention and let's make this a memorable evening together. Thank you. अरे अभी तो चाय भी गया
मैम शाम को 10 से आणि मैम संजय राउत ट्रांसपोर्ट सेक्टर
ठीक है